A restful night. Since I'm happy to see that the room is at least 1/3 full, with 5 minutes to go. And our keynote speakers here, our plenaries are here, and just to check before we start, is Corinne Yvonne here? Corinne Yvonne? Okay, so let me just check if she's gonna be online. So we'll be starting around 9 o'clock. I'll just come and do the wrap up with about 2 minutes to 9, and then we'll start the event. All right. Good morning, everyone, once again. I think I heard one person say good morning. Good morning, everyone, once again. Okay, at least I know that the room is full. Well, has people. So we're about to start day 2 of this 5-day conference. And as I mentioned yesterday, I believe yesterday was a good start to the conference. We started off with quite a show, which basically summarized our 5 years and had a very inspiring keynote from Emeritus Professor Ernest Aite, talking about the dearth of research in Africa and then the promise of what's to come. And we wrapped up with, I think, what I found were quite stimulating talks. And we hope that that was just the beginning and we're just going to keep going on till we hit the crescendo on Friday. So our first keynote of today is, he was one of our PhD, I believe when I started here at WACCBIP, he still hadn't defended his thesis. So he's one of our PhD products. And him along with Elvis, but primarily him, a lot of his work is what built the hearing impairment work that we've done here at WACCBIP. And he went on to do a postdoc at UCT, University of Cape Town, and has followed that up with another postdoc at the NIH. And when we thought of doing this, I thought it was only right that he be one of the keynote speakers because he's an accomplished scientist. And without much ado, I'll introduce Dr. Samuel Mauli-Adade to give the first keynote of today.
Thank you very much, Dr. Kwashi, for that kind introduction. It's good to be home, where it all began. My journey into hearing loss research began here in WACCBIP, and I'm so much happy to be here today. Before I start, I want to use this opportunity to thank the faculty of WACCBIP, the WACCBIP machinery, and everyone who is involved in training students here for the training I received when I was here. And also to thank the organizers of this conference for a job well done. I want to share one fun fact before I go into the presentation. So during our comprehensive exam, before our comprehensive exam, we all wanted to maintain the high standard and quality of science that is practiced here in WACCBIP. So we're all working hard. I remember one night, for the first time, I took several bottles of one energy drink called Rush in Ghana. And fortunately, it was 3 days to the exam. The exam wasn't the next day. And for a whole 24 hours, I wasn't able to sleep. From that day, I've never taken Rush again. So over the past 7 years, over the past 7 years, I've been working on hearing loss with the overall aim of identifying candidate genes and also understanding the pathobiology of hearing and look for variants that are population-specific with the aim of developing population-specific diagnostics for hearing loss. So today I'll be giving you a talk on what I did during my PhD. So hearing loss has been on the rise for some time now. WHO estimates that about 1.5 million people are currently living with hearing loss. Then in 2050, about 10% of the world's population would have some form of hearing loss. That's the estimate from WHO. When you come back down to Ghana, from our population and housing census, about 1.7% of the population are having some form of hearing loss. So we consider it as the silent epidemic. When you look at hearing loss, you can classify it based on the part of the ear that is affected. When sound travels through the ear, it goes through the ear canal and hits the tympanic membrane, which is the eardrum, generates vibrations which are carried into the inner ear. In the inner ear, you have the vestibular piece, which is responsible for spatial orientation and also balancing. Then you also have the cochlea. The cochlea is responsible for hearing. So you can hear me now because of the cochlea. And we have a lot of genes that are expressed in the cochlea to help us hear. Now, when the inner ear is affected, it results in sensory neural hearing loss. And when the outer ear or the middle ear is affected, it results in conductive hearing loss. It's easy to, I won't say easy, it's much more easy to cure sensory neural hearing loss. Sorry, to cure conductive hearing loss. So now that we are all here, if you cover your ear with your hand, then you can feel what conductive hearing loss feels like, because you cannot hear me very well. And through surgical procedure, it can be corrected. We also classify hearing loss based on the threshold of sound that an individual can hear. And here you'd see a graph. It's called the audiogram. We use the audiogram to examine the threshold of hearing of an individual. On the vertical axis, you see the various hearing thresholds, and on the horizontal axis, you see the frequencies that are tested. And on this graph, I have images to help you appreciate the level of sound produced at each threshold. So based on this, we can classify hearing loss as normal, people having normal hearing, then hearing loss as mild, severe, moderate, or profound. So I'll be using these terms as we move on. And also permit me, I'll be interchanging hearing loss and hearing impairment. They are all the same. Hearing loss can be caused by environmental factors. Specifically, we can have sickness such as meningitis, responsible for hearing loss. We also have indiscriminate use of antibiotics, such as gentamicin, resulting in hearing loss. Accidents can also cause hearing loss, and especially in the aged population, sometimes noise pollution can cause you to lose some of your hair cells and have some level of hearing loss at some frequencies. There are genetic factors that are associated with hearing loss. We classify the genetics part of hearing loss into two. We have the syndromic hearing loss that's linked to known syndromes, and non-syndromic is not linked to any syndrome. With the genetics of hearing loss, the genes that are associated are well studied in other populations, but when you come to the African population, we really don't know what are the main drivers of hearing loss in the African population. Here, we try to put a map of the various genes that have been studied. And on this map, you see that a lot of the sub-Saharan African countries don't have any study on hearing loss. And the gene that is mostly studied across the whole world is GJB2. GJB2 codes for gap junction protein. And these gap junctions form junctions between cells and allow cell-to-cell communication and help us to hear. So the common variants in GJB2 are, when we map them on the African map, you see that they are mostly in the North African countries, and sub-Saharan Africa is almost empty. And there is a founder variant reported within the Ghanaian population, and this variant, I'll talk to you more about it as we move on. But this makes Ghana to be a good place to study this particular variant. A lot of the studies in Ghana are focused on the environmental causes of hearing loss, with only 2 studies that were on the genetics. The first one from a village called Adamurabe, which is known for high prevalence of hearing loss, and another one was a nationwide study in 2001. Then our study followed up in 2017. So we had a fallow period where no one was interested in the genetics of hearing loss. So we sought to identify or determine the environmental causes of hearing loss and also the genetic causes. Then design population-specific tools for screening this condition within the population. Our sampling strategy is such that we go to the schools for the deaf, identify patients who have hearing problem. We classify them into either prelingual or postlingual. Prelingual, they develop the condition before the age of language development, and postlingual, they developed after the age of language development. We group them into two. We group them also based on the number of people affected in the family. So if you have more than 2 people affected in the family, we put you into the familial cases. And isolated cases, we're able to get only one person is affected in the family. Then we took samples from the healthy controls too. We extracted DNA and used next generation sequencing and targeted sequencing to look for variants. So this is a picture showing us our sampling in the school. When we go to the school, we obtain administrative clearance, then invite parents to come. We sign informed consent. After that, we do audiological assessment for the participants, both the hearing and hearing impaired participants. After that, we take blood samples. We want to sample at least 2 unaffected people from each family. But when we invite them to the schools for the deaf, they usually would send an elder sibling or one parent will come. So we have to travel to different parts of the country to recruit the rest of the family members. Before the WACCBIP van came, we used to travel in this kind of vehicle, which looks like a death trap, to very remote parts of Ghana to sample families. So what did we see? From the students that were in the schools for the deaf, majority of them have prelingual hearing loss, meaning they developed the condition before the age of language development. And a lot of them had their first comprehensive diagnosis between the ages of 6 to 11. So this means that the parents knew that something was wrong with their kid, but they don't have the center, or they don't have the resources to test them until they are ready to go to school. It's mandatory and compulsory for them to get tested before they're admitted in the schools. And the schools some of the schools have the facility to test, so they get tested when they are going to school. Then when you look at the cause of hearing loss, a lot of the prelingual cases are congenital, which means they were born with a condition. But when you look at the postlingual, they have illness such as meningitis, cerebrospinal meningitis, causing the hearing loss, and they also have convulsion. With the convulsion, the parents don't know what caused the convulsion, but they knew that their child convulsed and became deaf. Then meningitis, we compared our data with what has been published in Africa, and meningitis remains to be a major cause of environmental hearing loss in Africa. But when you look closely to the story of Ghana, you would see that in 1988, you have high proportion due to meningitis and also due to measles. And this reduced in 2019 when we conducted this study. And this is good to highlight because it shows that our health system is working and we are able to give immunization to kids and prevent some of these diseases. So now let's look at GJB2. I told you I'll tell you a story on GJB2. So GJB2 is part of the family of proteins called connexins. And the founder variant that was identified within the Ghanaian population in high frequency is not exclusive to Ghana. So on this map, I'm showing other places that the variant has been reported. And I'll talk about the Japanese population in the next 2 slides. So Ghana, because we have the high prevalence of this variant, Ghana is the best place to study this variant. So we looked at our cases based on how we classify them. And the familial cases seems to have higher prevalence compared to the previous studies. This is because the previous studies conducted used random sampling, but ours was more purposive. So we had higher numbers. But for the first time, we reported a carrier frequency within the population to be estimated at 1.4%. This means that these people within the normal population have the variant, the founder variant. They are hearing, but if two carriers get married, then they have the possibility of giving birth to a child with the condition. To help solve this, we designed and developed a diagnostic tool, and this was based on restriction fragment length polymorphism. We used the restriction enzyme NCI1, which is able to recognize the DNA and cleave at specific points and give us this kind of band pattern for genotyping. So we went back to the village with a high prevalence of deafness, sampled from there, and used our tool to test. We compared our results with Sanger sequencing, and we found out that our tool was very specific, and it's good, and it will be able to help us to genotype deaf people within the population. I want to say here that knowing that a quarter of the families we studied have this variant, it means this tool will be able to help us screen at least a quarter of the cases that we have, and it's going to be cheap because it's based on RFLP, which is not as expensive as sequencing. Knowing that the founder variant is present in other populations, we wanted to see whether we have the same variant, from Ghana, traveling all the way to the other populations, or it occurred independently in the various populations. So from the Japanese study, they estimated the age of the variant to be about 6,500 years ago. So we also decided to estimate the age of our variant from the Ghanaian population. And our estimate was about 9,600 years ago. So it's older in the Ghanaian population than the Japanese population. So did this variant travel from Ghana to Japan? We use haplotype analysis to compare the haplotype across different populations to see whether we have the same haplotype. And the haplotype in Japan and other population is different from the haplotype from the West African region, and also the haplotype from the Ghanaian patients that we had. So it's clear that possibly this event was independent, and it occurred at separate times independently of each other. We also identified a variant that results in a stop codon in the GJB2 gene. And this variant has been reported in Mexico. So I did not need to do the protein modeling. The model was already published by these guys in Mexico. We screened other connexins. We looked at GJC3. We also looked at GJB6. We did not find any variant, but we found a pathogenic variant in GJB4. And for us to examine this variant and see whether it might or it may account for the hearing loss in this particular family, we did what we call the protein sequence alignment. And when you do the sequence alignment, you are looking for the conservation at that position that you are studying. So here we are looking at position 119, and you see that the amino acid at this position is conserved in all the organisms that we studied, which means that position is important for the protein structure and also for the function. So we modeled the structure, and we saw differences in the wild type and the mutant structures. Now, let's look at the next generation sequencing part, the interesting part. I want to say here that we have a lot of stories that we can tell here, but I will tell you only 2 of them, because we won't leave here if I want to tell you everything about this part of the work. And forgive me, the slide is busy with a lot of diagrams, but I'll walk you through briefly. There's families that were negative for GJB2. We had 51 of them. We whole exome sequence them. So we're doing this in a tier kind of fashion, where we'll do targeted sequencing first, then next generation sequencing, exome sequencing, then the ones that we were not able to find a variant, then we do the genome sequencing. So with 51, we were able to solve 95% of them, combining both the targeted and the next generation sequencing, and this is very high. I think at the time that we published this, we were the first to have this kind of high rate of solving families with hearing loss. We found, 26 of our families segregated with non-syndromic hearing loss genes, 1 segregated with syndromic hearing loss. I'll tell you about this one. Then 13 of them had genes that were both non-syndromic and syndromic. We found 7 novel genes, and when we found these 7 novel genes, we looked for their expression in the inner ear. 6 of them were expressed, all of them were expressed in the inner ear. 6 of them had models, mouse models, and the phenotype was deaf. So being the first time reported within human, it's made it easier for us that we don't have to go the length of deleting this gene and doing the mouse study. So these are the genes plotted on the Ghana map, and it's very heterogeneous. Almost every family had its own unique variant, but there are 3 genes that were prevalent in the population, and this is myosin 15, cadherin 23, and GJB2. These 3 genes accounted for about 63% of the families that we studied. Then myosin 15 and cadherin 23, from our collaboration with our partners in Mali, Senegal, Morocco, and not Morocco, Mali, Senegal, and Cameroon, myosin 15 and cadherin 23 remain a common gene that is going through all these populations. So if we want to develop an African-specific panel, we will need to include these 2 genes. And we also have a North African panel that is good for North Africa, but we need to get a panel that would be for Ghana or West Africa as well. And I highlighted over here a gene in yellow. This gene is otoferin. This is the gene that is now in the clinical trials. And from our last meeting, we have good results from the trial. So probably our families in Ghana will be able to benefit from this clinical trial when it is done. The common syndromic deafness, except for thyroid stimulating hormone, which was high in the proband. The second story I want to tell you is on SLC12A2. This gene is expressed in the SLC12A2. It's expressed in the stria vascularis. And it's reported to be associated with this syndrome, Kilquist syndrome. And the exon 21 is important for the function of this gene in the inner ear, such that when you skip exon 21, it is not expressed in the inner ear in mouse and also in mammals. So we mapped all the variants that were reported in SLC12A2, and the ones in black are the ones associated with hearing loss. And you find that a lot of them are within the exon 21, which means this exon 21 is very important. And the variant that we identified in the Ghanaian population is also in this exon 21. Here, this is a family that had the SLC12A2 variant. And when you look at the audiogram, all the affected people have from severe to profound hearing loss, and the unaffected people are normal. They have normal hearing. At the same time that we found this, our collaborators identified a family in Pakistan. So we collaborated with them, and this is the Pakistani family. All the affected people still had severe to profound hearing loss. So we went ahead to model the structure for this particular protein and found differences between the wild type and the mutant structures, especially when it comes to hydrogen bond formation, which is shown here on the slide. We did functional assay, expressed the gene in HEC293 cells. The expression data shows that the mutants had higher levels for SLC12A2 expression compared to the wild type. And we think that the overexpression of the mutant would stress the cell. So we went ahead and assayed for phosphorylated P38, which measures the stress level of the cell. And it shows that the mutant cells were stressed and they were dying due to the overexpression of the gene. Then with the cell morphology, the wild type had this axon-shaped cell morphology, which was absent in the mutant. So we think that our variant is affecting the structure of the protein and also the function of this protein. So to summarize everything, the major contributor to environmental cause of hearing loss in Ghana is cerebrospinal meningitis. And 3 main genes can be put into a panel to solve about 63% of families. And these are GJB2, myosin 15A, and cadherin 23. And I want to mention here that all the sequences that we had, we've submitted them so that we would help solve the problem of underrepresentation of the African genome in the genome database. We've also submitted all our variants to CleanVar and CleanGene to help improve the gene pair curation and also gene phenotype curation as well. We designed a tool that we can use to screen hearing loss within the Ghanaian population. Then we wrote a policy paper to help guide policy in Ghana. We have newborn hearing screening program in Ghana, but we don't have the genetics as part of it. So we are suggesting that they include genetics screening as part of this newborn hearing screening program in Ghana. And the tool that we developed will be very handy here. So I want to spend the last few minutes to talk to you about my work in the NIDCD at the NIH. So in NIDCD, I work with 2 groups, the Friedman Lab and the Hall Lab. And in these groups, I work on proteomics and immunoprofiling of patients with hearing instability. So these are patients who have fluctuations in their hearing, and we really don't know the mechanism. So I'm working on that to find a mechanism that results in fluctuation in their hearing. And this is a common phenomenon in the American population, but not in the African population. Then I'm also working on a mouse model, and this is built on a variant that was identified in the Pakistani population. So I want to understand the biology of, or the mechanism by which this gene causes hearing loss. And also the overall goal is to develop a gene therapy for that particular gene. I want to thank my supervisors for the mentoring and thank all the head and staff of the hearing of the schools for the deaf in Ghana, and also all the participants, the parents, the students who gave us their samples. They trusted us with their samples, and we are very much, we appreciate their effort. So the next phase of the project is to is to get the results we have, to feedback the results to the community. And the team in South Africa, working with the WACCBIP communication team and the WACCBIP team, is trying to find the best way of getting back the results to the various families that were sampled in Ghana. Thank you.
Thank you, Samuel. So I think we have time to take quite a bit of questions.
Morning, great talk. It's interesting that the children with hearing impairment, they have blue eyes, right? Is there any link with, high color genes and their hearing genes?
Okay, thank you so much for that question. So with the blue eye, the gene that is responsible for the coloration of the eye, the pigmentation, it also has something to do with hearing. So the same gene, when mutated, affects the eye and also skin. That's how come we see some of the pigments on the skin also losing, some of the cells in the skin losing their pigments, and also the premature gray hair. But to broaden your question, the eye is connected to the ear as well. So the vestibular part of the ear helps us to balance, and it works with the eye. So when you close your eye, you don't have 100% balance. And when your vestibular piece is also not functioning, you can also not balance very well, even when you have your eye open. So they work hand in hand.
Thanks, Samuel, for the great talk. Awesome talk. Good friend. I was wondering the plans for your clinical trial.
Can you say that again, please?
I was wondering the plans for your clinical trial.
The clinical trials.
Yes.
Plans for clinical trials. Yeah, I think we will get there, but we are not, we will do the animal studies first. So we do animal trial first. When we are done with that, then we can move into clinical trials. But clinical trials for hearing loss is quite expensive to see. So we will get there.
Okay. Thank you very much for your talk. I think you show clearly in your data that, meningitis is the main cause of loss of hearing in your study in the young people. So I'm wondering, did you try to also investigate in, let's say, adult people, mainly in old people? Because, sorry for my ignorance, it looks like when people are growing, when they are getting old, they lost also hearing. So did you try to investigate what is the cause of loss in hearing in this population? Thank you.
Yeah, thanks for that question too. Yes, so in the future, we have plans of doing age-related hearing loss. But currently, our focus is on the pediatric hearing loss, because if we are able to diagnose early and give the correct intervention, we will be able to help them with their cognitive development. And there is one thing I did not talk about it in this presentation because of time, but what we found in the Ghanaian population in Mali, Cameroon, is that these children with hearing loss are born to hearing parents, and the parents are not able to communicate with them. And we don't even have infrastructure. Our schools are not built for the hearing impaired. So it affects their quality of life, it affects their relationship with their families, and so on. So we are much more focused on the pediatric first, but in the future we'll be looking at the age-related hearing loss.
Okay, Dr. Dinko.
All right. Thank you very much, Samuel. Very nice talk.
My question is with regards to the time of development, how you determine the time of development of hearing loss, whether it's postlingual or prelingual, because at a certain point, the people concerned cannot actually hear. Did you ask the parents or you did a test? The second question is, did you find any relationship between this time of development of hearing loss and the severity or the level of the hearing impairment? Yeah.
Okay, thank you. So with the prelingual and postlingual, so we look at people who develop the condition before age 2 into prelingual. And actually, there is another phase between the prelingual and postlingual, which is the perilingual. That is between 3 to 4 years. But we combine perilingual and postlingual together to be postlingual. So anything from 2 years above, we put into postlingual. Yes, so the by age 2, the child should be able to make some sound to call mama, papa, and things like that. So some parents or all the parents know that my child was not able to make these sounds, will not respond to sound before this age. Oh, no. So that helps us to classify them. Then your second question, whether there is any link with severity. With what we noticed is that people with postlingual hearing loss tend to learn how to speak because it's postlingual, especially those who develop the condition after the age of 10 years old. There is one particular patient that I sampled, and she did not know she was deaf until she was in high school. And her uncle came home complaining that, I met you in town, I was calling you, and you think you're now of age, so you can be arrogant. Then she said she couldn't hear. So it was after then she began to take note that when people are talking behind her, she cannot hear. So she's able to read the lip, but not able to hear sound. So that's what happens with the postlingual.
Okay, thank you. I think we have a question online. It will be projected on the screen from Dr. Megache. So apart from the genetic mutations of connexin, especially GJB2 and its variants of the inner ear, are there some genes affecting outer ear defects? And can other factors apart from genetic contribute to hearing loss? Well, I think.
Yes, please. So there are other genes that are affecting the vestibular part. So people who are studying balancing and spatial orientation of the head are able to come up with genes that affect the vestibular part of the ear. Both the vestibular part and the cochlea, they have hair cells, but the hair cells are connected to different neurons to the brain, and they perform different functions. So we have genes that are expressed in the vestibular piece. And yes, we have different causes to hearing loss. We have the noise-induced hearing loss. We have ototoxicity, which is due to use of drugs like anti-cancer drugs and antibiotics. We have sickness or accidents also causing hearing loss.
Okay. Okay, so I think we'll take one quick question from Chinedu and one more from Nikes, and then we'll move on.
Okay. Thank you, Samuel, for the presentation and for the work you're doing. I just have two questions. One is on the mechanism of hearing loss and pathogenesis. Is it the same regardless of whether genetic, environmental? Is the pathogenesis the same? Then the second question is on the gene, the GJB2. Is the gene only functional or associated with hearing loss? Does it perform any other function in other parts of the body? And the protein, so when you look at the protein, what happens? So is the protein not expressed, or the defect is only at the genetic level? Thank you.
Okay. So the mechanism for environmental hearing loss and genetic, they are different. They all affect the ear. Even with the genetics, different genes have different mechanisms to which they affect hearing loss. So, for example, someone who has noise-induced hearing loss might have some of the hair cells dying at certain frequencies. And this is also common to orthotoxic hearing loss. But someone who suffers an accident can have the bones in the inner ear displaced, and that can also cause hearing loss. With the conductive, some children are born with a flesh covering the whole ear. So they have to do surgical procedure to remove the flesh for them to hear. So the mechanisms are different. Then with GJB2, yes, so the expression, I would say that depending on the variant, some variants result in reduced expression of the gene. Others will not express the gene at all, especially the truncated ones. You won't have the protein to perform the function. Some of the variants, you have the protein all right, but the protein is not functional because it's supposed to form gap junction to allow for cell-to-cell communication. But if this junction is not formed, then the cells cannot communicate.
Yeah, very nice talk. Yeah, my question was just to expand the previous questions. So there is a growing interest now in the North to explore epigenetics in hearing loss and many non-communicable diseases. Do you have plans to explore, to extend and investigate that in Africa?
Yes. Yes, so the study is still continuing. We are still looking at patients, and we plan to extend the study. So the collaboration between, this work is a collaboration between WACCBIP and Hygienes Africa, and the collaboration is still going on, so we'll still be doing more.
I think we can all agree that was a great start to day 2. And to continue on from there, I'm going to invite the chairs for the next section, which will be molecular epidemiology and enhanced epidemic preparedness. And these will be our very own Dr. Elma Amatepe and Dr. Lucas Amenga Etigo, and they will take us through to the next session.
Hello everyone, good morning. It's good to see you all this morning. So far we've been making some very good time. So at this point, we'll hope and ask our next speakers to emulate our keynote speaker and try and keep to time. First on the list we have Chinedu A. Ugu, and he's going to talk on the pandemic preparedness post-COVID-19 in Nigeria, African Rapid Immunosurveillance System for Epidemic Response Arise. So Chinedu, the floor is yours.
Good morning, everyone. I just hope I can keep up to time. That's the problem.
Try.
So I'm going to tell the story of African Rapid Immunosurveillance System for Epidemic Response called ARISE. My name is Chinedu. I'm a senior lecturer at Redeemers University and a postdoctoral fellow at the African Center of Excellence for Genomics of Infectious Diseases. And I'm currently at the NIH as an APTI fellow. So I'm going to start with telling the story of ESGID. I guess most people, if you don't know, so ESGID is the Center of Excellence, more like the sister of WACCBIP in Nigeria. So ESGID was also founded in 2014, so we'll be also doing our own anniversary this year. So SG, we were very instrumental during the Ebola outbreak in 2013-2014. And then for SARS-CoV-2, we provided the first genomic sequence of SARS-CoV-2 from the continent. And so far, we've sequenced over 10,000 SARS-CoV-2 genome across 26 countries in Africa. And currently, SGID is a reference lab for genomic sequencing for COVID-19. So the story of ARISE, this is the summary. So in 2018, I was finishing my PhD. I moved back to Nigeria with support from the BBSRC on a project called One Health Accelerating Vaccine for Ebola and Lassa Fever. This was a collaboration between SGIT and Nigeria, and the aim of the project was to study immune responses to Lassa fever in Nigeria. So while we were doing that work, then COVID happened in 2019, and so most of the capacity we built with Lassa fever, we translated into working with COVID, and which we got a good seed funding from Cambridge, Africa, on the project SARS-CoV-2 Immune Correlate of Protection in Nigeria. Then with that effort, we also got some good support from the Gates Foundation to then set up the actual the main project, which is what I'm going to talk about, the Africa Rapid Immunosurveillance System for Epidemic Response. So the main objective of the ARISE platform is mainly, one is with the enormous progress we've made in genomic sequences, so it's more like providing immunological data to support the genomic sequences. And then we hope that most of those immunological data we'll share with public health agencies like the NCDC in Nigeria, the Africa CDC. And then secondly, one of the things we found that was a struggle during COVID-19 is having curated samples. So another aim of the ARISE platform is to curate sera PBMC from mostly emerging pathogens that you have endemic in the continent, and then keep them, should in case there will be any pandemic or epidemic, we'll provide this sera for research and for interventions. So also we are hoping to be like, to be a center of excellence for immunological research and then training. And then all of this data we generate, we ensure that we share them in real time with all the public health agencies. And then to mention here, the whole essence of the ARISE platform was built on collaboration, both within the continent and then outside the continent. So this is what the platform looked like. So we have antibody assay where we use both ELISA and then Luminex. And then we do, to measure binding antibody response, we use viral pseudotype neutralization to measure neutralizing antibody response. We use ELISpot to measure T-cell functional assay, and then we use flow cytometry to do phenotyping. And then we also do single cell and B-cell and monoclonal antibody discovery using RADpig. So I'm going to start with two examples of the work we've done with ARISE. So starting with Lassa fever. I don't know if people don't know Lassa fever. Lassa fever is one of the viral hemorrhagic fever that is endemic in West Africa. So it's one of the old world arena viruses. It's genetically diverse. There's no licensed vaccine for Lassa fever, but there are two vaccines that are currently in clinical trial. What the gap in Lassa fever is that it's one of the neglected tropical diseases. So the immune correlate or the features of protection is not yet defined for Lassa fever. So one of the work we did is with the OVO project is to define some of this, the type of immune response you see from patients with Lassa fever. So the first thing with some of the data, one is here, we measured T-cell response from Lassa fever survivors in Nigeria, and then the contact. And these exposed contact are the people who manage Lassa patients. So you could get Lassa from contacts and also from the rodents. So we recruited survivors of Lassa fever and their contact. And these contacts include those who take care of them, the family members, the clinician, and the nurses. And the interesting thing we see, measuring their T-cell response, and here we use the nucleoprotein of the virus and then the GP as the antigen. What we see is that both the survivors and the contact who never tested positive for Lassa fever, we are making the same level of T-cell response. And also we could show that the binding the major T-cell response is coming from the NP1 region of the nucleoprotein and then the GP2 region of the nucleoprotein. We also measured the binding antibody response using ELISA, and again, what we saw is that those who didn't even test positive for Lassa, which are the exposed contact, and then those who tested positive, again, had similar level of response. So, but where it becomes interesting now is when we then did the neutralization assay. So we have pseudotype viruses that expresses the different GPC of the different lineages of the Lassa virus. So we have lineage 1, 2, and 3, and 4 of the Lassa virus. And then what you then see is that it's only the survivors who make neutralizing antibody response, while the exposed contact do not. And this is simply because the survivors have symptomatic disease. They are clinical cases, while the exposed contact, they are asymptomatic. So we think they don't have enough time to make neutralizing antibody, even though they already have the binding antibody response. So then, when COVID happened and then we got funding, we basically translated everything we've done for Lassa fever COVID to understand the immunological responses from different COVID cases. So for COVID, we recruited the different doses of the vaccine cohorts. And then we also have the convalescent people that survived COVID as well. We also have the exposed contact. One special population we have is the pre-pandemic samples. So these are the samples we collected from Lassa fever. And then when COVID happened, we also tested them. And then we have the breakthrough, and then we have the non-negative control. And these are the people we've taken time to create and ensure that they are negative for most of the endemic pathogens we have in the continent. So for COVID, the take-home from our COVID assay result is we also did binding assay and then neutralizing assay. And what you see with COVID is that both the exposed contact and then the survivor, they make the similar binding antibody response, and they make the same level of neutralizing response. And this is, I think here we tested the different strain, the Y type, the beta strain, the Delta, and then the Omicron BA1 at the time we did this work. So different from Lassa, COVID people, whether you were asymptomatic or symptomatic, you do make both binding and neutralizing antibody response. And then this is one interesting thing we found, and which we hopefully other people also have reported it, is that the pre-pandemic serum, these were the people that were samples we collected pre-COVID. And you see that they also have the same level of binding antibody response with both the convalescent COVID-19 patient and then the contacts. But the interesting thing about this is that this pre-pandemic serum, they didn't have neutralizing antibody response. They only had binding. So this probably will be from the old coronaviruses that is different from SARS-CoV-2. So this is the summary of everything I will tell you. So the key thing we'll do, like I said, we generate all of this immunological data and we'll share them in real time. But one thing we try to do is, again, to curate some of these samples, both the PBMC, the sera, should in case there's any outbreak. I think so far we have about 540 samples from Lassa survivors and then about 650 from SARS-CoV-2 survivors. The other part I'm doing now is I'm currently at the NIH, and my project at the NIH is mainly isolation of monoclonal antibodies from these Lassa survivors. So most of these samples that we've created, so the aim now is then to isolate monoclonal antibodies from them that you can then use for diagnostics and therapeutics. And again, about the training, so far from 2018 to now, we've graduated about 8 MSc students and then 12 undergraduates. So those are some of the outputs from the work we do with the RIs. Then the current work ongoing, now we've expanded the RISE program, and we now have also other funders beyond the GATE and then the Cambridge Africa. So we have different projects ongoing. Like I said, I'm at the NIH doing the isolation of the monoclonal antibodies. These are some of the PhD students we have at the NIH, at SGIT, and some of the master students working specifically on the RISE projects. So we have other projects on Lassa fever, which we are trying to understand the bottleneck between transmission. So bulk of the Lassa fever infection comes from rodent to human, but some cases in the hospital where you have hospital transmission, nosocomial infection. So what we want to understand is why those pathogens manage to do human to human versus the normal route of rodent to human. So we have a PhD on that. We have another PhD student who is now looking at the correlate of protection from Lassa fever. We also have other viruses that we study. We're looking at epidemiology of HIV, HPV infection. So these are some of the ongoing work on the ARISE platform. So I won't say all of this without telling you the challenges. So I could say, oh yeah, everything worked out perfectly for us, and then we are doing very well. But no, we also have our own successes. So one is funding support. You also struggle to get good funding. And then one of the challenges, and we tried our best in correcting it, is where when you do collaboration, where people are more interested in you sending samples to them without building capacity. And that is one thing I think we've made progress with, ensuring that whatever collaboration we do is either proper benefit, is beneficial to both team, both the collaborator and us. And the other challenge we talked about, and I think everybody will have this problem, is lab supply. By the time you get samples from the whole, by the time you get samples to work, it may take 6 months to get the reagent to do the work. And then I don't know if you guys also have this problem, but retention of talent. In Nigeria, we call it the Japa syndrome, where every young, talented PhD student wants to travel abroad. And I don't think I should stop them from going abroad, but I do think we should find a way to, whatever is attracting these young people, talent to abroad, we should find a way to retain them here, and so that they will also continue the legacy of doing excellent research. So I think this should be my last slide on this. I'm going to thank all the funders and all the supporters we've had over time. First of all, my supervisor in Cambridge, who has enormously supported me and ensured that I set up, and also my director at SG, Christian Happy, and the funders, Cambridge Africa, who believed in us when we think we're not doing anything. And then the Bill and Melinda Gates Foundation. I also will thank the NIH that is helping to then take us to the next stage, which is translational immunological work. And every other person that we've listed that I don't mention. So lastly, I will say thank you, and I could take questions. I just hope I'm keeping to time. But I will also use this opportunity to invite you guys to our own anniversary, which will be in October, and we'll be launching our new genomic center. So if you've enjoyed Ghana Jollof, don't worry. You know you have a good time coming to Nigeria in October, and I think at least now we'll take questions. Thank you.
I was going to commend your very good timing, but you had to bring in the Ghana Jollof. But that was very good timing and good presentation. Thank you so much. We'll take questions. Peter has his hand up. Yes, so Dr. Kwashi.
Hi, Chinedu. Great talk. And I'm happy that you finally agree with me that those cross-reactive antibodies are not neutralizing.
I've always agreed with you.
Okay, so I had a question about your Lassa samples where they are asymptomatic. Do you think it's because there wasn't enough viral load in the initial infection to actually have a productive infection, so the immune system is exposed but not really, the virus is cleared before it actually starts anything? Is that what you think is happening?
Everything you say is correct. No, we don't know what is the actual mechanism, but that is one is probably they didn't have enough viral load. Or it could also be true they are genetically diverse, that they're able to mount a good response. So I don't think I mentioned it, but I could say that in Lassa fever, 80% of the infection are asymptomatic. Mm-hmm. So the problem is that the 20% that are symptomatic, the fatality is very high. So that's why it becomes a big problem. So when you look at this, a lot of people will carry antibodies. So you say that, one, you're correct, that the infection is not enough to have mounted a good immune response. So those people will survive, or they have good immunity that they were able to clean up before it becomes symptomatic, or it's genetic. So the answer is we don't know.
Hello, Chinedu.
Yeah.
Thanks for the presentation. So, how did the immune response play out in COVID, knowing that Lassa is already existing and there could be comorbidity with COVID? Did you look at that?
That's one of the things we want to look at. So you see what that was the idea we want to test those Lassa samples. So we first of all thought, okay, this is endemic, and then COVID happened. Do we see anything? And then which is when we saw the binding antibody response. Oh, okay, people already have this. So we then looked in neutralization, we didn't see anything. So part of the thing we intend to do is to go back to our Lassa cohort, because that's one good advantage we have with SGIP, is that we have a clinical site supporting us. So to go back to our Lassa cohort and see those of them that had
COVID alongside Lassa and how they fared. So those are things we want to do in pipeline. Thank you.
So thanks, General. I enjoyed your presentation very, very much. And I think you have a treasure trove, and you mentioned at the tail end that the focus of your postdoctoral work is actually to try and, isolate monoclonals. And my question is, what are your plans now to exploit this treasure trove and make diagnostic products and therapeutic products? It's very exciting to me to see that that's part of the plan.
Well, you are my plan, Oh, okay, thank you. I think you are my plan. So it's a good, so for us, again, the end game or the major objective for everything we do with immunology is to get to the level of translational. It's not just how do we get this to the people? So part of the work I'm doing at the NIH is to isolate the monoclonal antibody, and then hopefully once we get them is to do clinical trial and see how we can get them into the market. There is no, like I said, there's no licensed vaccine for Lassa. And one of the set of people that are very endangered or vulnerable to Lassa fever are the healthcare workers, the clinicians. They are usually the most affected. So if we can manage to get a therapeutic antibody that they could take, that they can, when they handle some of those patients, I think that will be a game changer. And I think this is what I'm hoping to. Once we get this monoclonal antibody at NIH, which we are making progress, it's just that the NIH is not part of the presentation. So once we get this antibody, is how to then get them to the people. So you are part of the plan,
Yeah. All right. Yeah, thank you. I have two questions. Yeah. One question is, I want to know which variant or which lineages you use for the neutralization assay for the Lassa. One. The second question is, can you elaborate on your collaboration with Enfio Liberia, please? Thank you.
So I didn't get the second question.
Your collaboration with Enfio. You mentioned Enfio as your collaborator in Liberia. So can you elaborate on that, please?
In Liberia?
Yes, you mentioned.
Oh, okay, thank
yes.
you. Yeah, sorry. So it's early morning. I don't work well in the morning. So the neutralization. So this is the interesting part that this is the capacity we are doing very. For this, the work I presented, we use the GPC of lineage 1, 2, 3, and 4. So that's what we presented. But I could tell you now that we do have capacity for 17 lineages for Lassa. So when I say 17, it doesn't mean that you have 17 lineages circulating. So some of them are not real lineages, but we hope that we should be able to cover both the existing lineages and those that will envisage that will happen in the future. So we have capacity to test all of them. But for the work I presented, we've only tested 4 lineages. So the collaboration we have in Liberia is with, I think you saw it at the acknowledgement. So that's with Exit. So that's the genomics work that we do. So we do support most African countries, Liberia, Sierra Leone, Guinea. So I think I won't be the best person to comment on that. Christian Happy will be the best person to comment on that. But those are the genomics work that we do. Thank you.
Yeah. Yes, yeah. There are more questions too.
Jesus.
Yeah. Yes. Yeah.
Right?
Thank you.
Yeah.
Yes.
So
I'm here.
Okay, there you are.
Thank you so much for the wonderful presentation. It's interesting to see the Lassa fever patients and their close contacts. You have similar antibody levels. Do you think the differences in neutralization ability is mainly ability? Yes. Yeah. So what I'm asking is, between your patients, the Lassa fever patients and their close contacts, you have similar antibody levels. I'm wondering if you've investigated antibody ability subclasses, maybe if that could explain why you have different neutralizing abilities.
I didn't get it. Or the subclasses of the different antibodies?
Yes. So
well, no, we haven't gone to that level yet. We haven't classified it. So what you are seeing here, the binding antibody is IgG. So it's only IgG that we've worked on. So we haven't looked at the different subtypes of the IgG yet. No.
Okay. All right.
But I do think, like, which is what Peter alluded, is why we do think is that the contact, even though they are not testing positive, they must have had the virus at some point. So they may have had asymptomatic cases. Their body do have this whole signature of immune response. Now, whether that immunity is what is protecting them or it's just there, we don't know yet. Thank you.
Thank you very much.
Next presenter will be our own Dr. Yawaniwe. See here.
It's online. Yeah. Can someone, Martin, check in the office? Maybe he's forgotten. It's communications here. If you can play some of our sponsors stuff that whilst we wait. 2 minutes. If you don't find him, we'll change. Video. Yeah. I don't even know. She's saying that. And then we are going to the fellow sessions. Oh. Communications, you guys.
Akwaba to all my lovely, lovely friends from Ghana and in and around. So thank you so much. I'm actually not from Ghana, as you can see, right? Well, we are all a big bunch. We're all a big bunch of scientific geeks that have come here to explore and learn and evolve. My name is Vikas, and I'm from Mumbai, India. And from India, I greet you all with Namaste. That's what we do, right? So thank you so much. I've traveled all the way from a city called Bombay. I'm sure Mumbai is a city that you guys probably heard of. It's about 8,000 kilometers away. And this has been a very exciting 2 days for me. I got in 2 days back. And let me tell you, I don't feel I'm 8,000 kilometers away. I feel I'm very much in my home, in like India, because the Ghanaian hospitality, the people of Ghana, really, you guys have touched my heart, touched the heart of every Indian. Thank you so much. Right. And from the time I landed into this beautiful country at the Accra airport, I was greeted with a smiling officer at the customs, and she smiled, and I don't know why she was smiling, and everyone, and everyone that I met has been smiling at me in Accra. So maybe I'm like the Tom Cruise out here, I don't know. But lovely people, guys. You guys are lovely people. Let's give a round of applause to all you people. So I'm actually dressed in the WACCBIP dress as well. I'm going to take this back and I'm going to show this to all the scientific community back in India. We have a big scientific community in India. and I'm going to show this to them. This is what Ghanaians do. This is how they make an event successful, right? So thank you. I'm not going to take much time. Of course, this is a very different presentation. It's about our company. It's about how we add value to science and research. And I'm just going to take a little time of yours, maybe 5-10 minutes. And if I run out of time, you can just tell me to stop and I will get out of here, okay? But it's a scientific conference. So apart from learning and absorbing different scientific research papers, it's very important for all of us to understand the value of the vendors, the people who provide reagents, the people who provide all the stuff for your laboratory work. And we're one such company doing this for the last 50 years, yeah? This is a family business. It's actually 47, I'm sorry for that, but this is a 50-year-old company. We're called SRL, Cisco Research Laboratories. We were the first company in India that started manufacturing molecular biology and bioscience products. The story goes like my grandfather was a very poor man back then, but he had a vision, and he was invited by the President of India to start manufacturing products, because the President of India told my grandfather, why should we import from Sigma, Merck, and Thermo? You know, that's the Indian story, right? We have a lot of respect for these European and American brands, but the president said, you know what, I will give you $500. He told my grandfather, I will give you $500. Why don't you set up a laboratory? And that's what we did, because we didn't have, my grandfather was a businessman. He didn't have money, but he had the vision to make a big company. And with $500 of money, today we are about $45 to $50 million revenue company selling products in more than 100 countries. So we started manufacturing with amino acids, and yeah. And let me tell you, we sell to more than 40 countries within the African Union as well. So we are a very big presence here. But anyways, I'm going to move ahead now. So our motto is the endless process of science. And what we do is we manufacture molecular biology, American Chemical Society grade, that's called the ACS grade reagents. We're obviously very strong in pharmaceutical and biotech products. As you know, India is a hub for manufacturing the formulations, the drugs, the vaccines. And our company provides raw materials and biopharmaceutical products to companies like Sanofi, to companies like Pfizer. They have huge operations in India. Of course, in Europe we sell as well, but we have a very big base of pharmaceutical products. There's only 1 or 2 companies in India that manufacture culture media for microbiology. So we manufacture our own dehydrated media. We do raw materials. We have animal houses. We have our own peptones. And we are the first company that manufactures DNA and protein tools. So all the COVID engulfed our lives in 2020, right? And as you guys have fought the virus here in India, we were doing the same. And our company was the first company to start manufacturing all the reagents which were used by the diagnostic companies that started selling of the kits. So we've played a very pivotal role in Southeast Asia to provide diagnostic reagents to all the kit companies, right? From Korea to China to India to even the Gulf states. So we have a company called, that's another division called BioLit, where we make a lot of Taq polymerases, our reagents for DNA and RNA extraction. Well, not going to explain much, but they're used for quality control, analytical development, and as I mentioned, pharmaceutical and biotech manufacturing, right? So this is a little bit about us. We have 3 manufacturing locations. In India, we have 5 distribution warehouses. We are a reputed company. We have the ISO 17025 certification, which is a quality certification. We are one of the few Indian laboratory chemical companies that have FDA and CGMP. There's not many Indian companies that do the FDA registration for lab chemicals. We have, of course, maybe more than 200,000, 300,000 scientific papers. We employ about 300 people on payroll, but about 200 people on contract. So in a nutshell, we're a company giving jobs to more than 500 people. And let me tell you another interesting thing here. For all the lovely ladies here, in our organization, all the top positions are held by women. You know, we have, yes, there's one manufacturing unit which is only controlled by women. Right from the sweeper to the head, there's only women. We do not allow even men to join the biotech unit, okay? That's our contribution to helping, giving equal opportunities to all the ladies of all, the scientists in our country, and that's what we do. Of course, as I mentioned, we are present in the complete world. We have about 800 global dealers that are serviced by 120 distributors globally. We have about 8,000 products. and about 20,000 stock keeping units. In short, guys, we're called as the mini Sigma Aldrich of India, because this is what the tag was given to us way back in 1975 when we were established. People called us the mini Sigma of India because we were the only company that had the quality that could match up, that could stand up to an American or a European reagent. And even today we are called as mini Sigma of India. That's why I'm here to present our products, to showcase our offerings out here. Of course, we sell for different purposes: semiconductor, biology, proteomics. You guys are doing a lot of research here in different departments at the UG and all the different places that the other students have come from. We are very strong in molecular biology, in even for organics like HPLC, gas chromatography solvents. We are the only company that manufactures nano powders in India. There's a lot of emphasis now on the increasing nanotechnology sphere. There's a lot of money being pumped in by different governments all over Europe, all over Africa, all over the Asian continent for innovative products, innovative drugs using nanotechnology. So we manufacture graphenes, we manufacture nanotubes, we manufacture lots of nano powders, which you could buy now from us since we are here. That's the idea. So of course, I'm not going to go too deep, but just a little run through of what we manufacture. We have a big range of HPLC solvents, which, any HPLC lab, they need the basic things like acetonitrile, methanol, N-hexane, water. We do a lot of these products. We do dried solvents. We do ion pairing reagents. We are very strong in organics. Of course, this conference is more to do with cell biology, but I'm just going to run through all the different products that we do. We have a big range of plant growth regulators, even precious metals, boronic acids. We are very strong in silica gels. I know you guys use a lot of silica gels even in the UG different departments here. We manufacture those in a very big scale. Let me tell you, we are the only company that have solvents which are molecular biology grade solvents. That means our products are DNase, RNase, protease free. I'm talking of solvents which are used for extraction purposes. Usually you would not find molecular biology grade solvents. So we manufacture the MB grade solvents which are DNase, RNase, protease free. They're very interesting. Very few companies in the world make molecular biology grade solvents, and we're one of them. Of course, biochemicals is a very big foray of products. We do various enzymes, substrates, detergents, sugars, amino acids. We are the pioneering company for molecular biology from India, and that's why the WACCBIP really interests me, because this center is all about biotech, diagnostics, molecular biology, cell biology. We have a huge range to meet all your different lab requirements. Electrophoresis. We are the electrophoresis leaders in Asia. I'm sure you do a lot of electrophoresis in your labs. We are the largest manufacturers of agaroses in Asia. We sell agaroses to companies like Sigma, to companies like Thermo, to companies like Merck. We actually sell our products to a lot of these companies. There's a slide that I'm going to show you guys after this. Of course, catalysts, DNA, RNA accessories, we have it all. As I mentioned, we are the pioneers of nanotechnology in India. We sell products for drug delivery systems, for research in different centers of research that are engaged in nanotechnology. Our medias are products that are used primarily for microbiology. As you know, you guys might be using products from companies like Oxoid, like BD, Diffco. We buy a lot of the raw material from the same sources in Europe where they buy it from, so our product quality is equally good, with, of course, a much, much, much more affordable price for our Ghanaian brothers and sisters. So, and I'm very sure about this, guys. We also supply to some of the labs here, and we would hope to have more business because we want to add more value to your work that you do. That's a little range of our protein tools. So we do reference standards, we do protein markers, we do columns. We have beautiful products. I mean, I'm going to actually have you guys distribute a little brochure so you guys can go to our webpage. Our webpage is like the Aldrich webpage. It's very advanced. There's a lot of information for all you students to learn and understand. So, of course, we are very strong in pharma ingredients. Of course, this is an institution, but institutions are the birthplace of industry, right? So there's lots of products that we do in bulk scale, and these are some of the specialty products. We do it in lab scale as well, but we do it in bulk scale as well. Vaccines has been a very, very big contribution from our company. All the big vaccine projects. I don't know if you guys have heard of an Indian company called the Serum Institute of India. The Serum Institute is the world's largest vaccine manufacturing company. It's bigger than most of It's bigger than Janssen. It's bigger than many of the companies in Europe. We are in a very strong partnership with the Serum Institute to manufacture, I'm going to tell you guys a very interesting story here. The COVID vaccine, okay? I think the COVID vaccine which we got, the Indian guys got, was made by the Serum Institute. It was developed by AstraZeneca in England. And it was a chimpanzee. I'm not a bioscience student, guys. I'm a business graduate, but I think last 15 years I have learned a lot. So excuse me if I'm wrong. You will not believe Serum Institute is actually developing the world's first vaccine for malaria. I think that vaccine is still in it's in the trial stage right now, but or maybe it has come out. I'm not sure about the what's the name? I'll share it with you. I will take my royalty for that from you. So the malaria vaccine was used. So what happened is November, December 2019 is we started hearing cases of COVID, right, from China and from other countries. Serum Institute already had a whole platform ready for the malaria vaccine, and they used the malaria vaccine platform to actually immediately divert their interest to the coronavirus vaccine platform. So if there was no malaria, there would be no COVID cure, for the Indian population, because we had 130 million people, 1.3 billion people in India. So it's very important that this company, the Serum Institute, is a very big consumer of our products, and we are very proud to be associated with all the different government platforms that help for COVID. In fact, the Fortune magazine in the US, they actually covered me as a COVID warrior in 2020 because we did some really good work. We tried to save lives indirectly, by developing kits for the coronavirus. So we're really proud that we got this opportunity. Well, a little bit about our labs. These are just our R&D labs, and I'm not going to go too deep in here, but this is some of the products, the way they're stored. We have more than 500,000 to 1 million bottles here that we stock. This is all our warehouses in India. We have 9 locations in India. We sell to more than 120 countries globally. And this is how our product looks. So our labels are pretty much like how you could have all the necessary information. Our entire family of products is here. This is how our typically our label, they look like, and we have all the information like specifications, impurity profile. The difference between a good laboratory reagent and a regular normal laboratory reagent is how many companies give you the impurity profile. Everyone will say it's a 99% or it's a 99.9, but the main thing is the impurities. And we focus on the impurities because we're a company that consider Fluca as our Bible. Flucarydaldehyde, I'm talking of 100 years back when, I don't know where I was, but my grandfather, he was the one who took up the Fluca book, and he devised every single specification of SRL to be in accordance with Fluca specs. That's our power. We are the gold standard in the lab reagent business. Well, as I mentioned, we have huge number of distributors. We have a presence in the African subcontinent. You could see the countries that we have representation in. In fact, I'm so much in love with Africa. This is my 5th trip in like 1 year. And I'm again actually coming to Cape Town after 2 months for another show called MedLab. It's a diagnostic show. But I've started to love African people. I've started to love the way you guys are trying to build your country. Very much a story like India. We are exactly like you guys. We really worked hard. But I can see that Africa is the future of the world. It's the future of the world. That's a fact of life. It is. And I think I would like to tell you that we have a lot of African students in India, by the way. There's a lot of African students researching in different research centers in India. So there's a lot of friendship of scientific knowledge share between India and African nations. So it's good. So these are some of our clients, guys. So like Dow, Abbott, BASF, Bayer. You can see the kind of companies that use SRL products. There's also global testing services like DNV, SGS. There's food companies like Coca-Cola, Diego, Heineken, Kellogg's, Kraft, McDonald's. We eat this stuff every day. We sell to all these companies. We actually sell to all the labs of all these companies. It's taken a lot of effort. It's taken a lot of convincing, but it has happened. Because, we all want a Mercedes and a BMW, right? But actually, the stuff, what is inside, like another car, which is half the price, is probably the same. But the BMW has the brand, right? So we're trying to build a brand, and we want to tell you that we are the brand for all your work. done, almost finished, some clients. These are some of the companies, guys, that we manufacture for. So you can see, I'm sure you guys buy your products from a lot of these companies for your lab work. And you won't believe these guys buy from us. It's a fact. I mean, there's nothing to lie about. So the many products they buy from us because they know us. We've been around for 50 years, as I mentioned, half a century. And our products, our quality is good. They take our products, label it into their brand, and that's how business works. This is how business works. The reason why I'm here, if you can buy and trust them, and if we are one of their vendors, why don't you can buy it from us as manufacturers, right? That's the whole idea. Because Africa, the same thing is you have limited resources of funding and you have good knowledge. So you got to save money somewhere, right? So we're there to help you save money. That's our objective, right? That's it. Thank you so much. Thank you. I'm really happy that I'm in your beautiful country. And 3 cheers for all Ghanaians. Hip hip, hooray. Hip hip, hooray. Hip hip, hooray. Thank you, guys. Thank you.
Sorry.
Champion Makata.
Thank you very much. I guess you all agree with me that this passes for a keynote lecture. This passes for a keynote.
I just want to add a global So he's our local distributor in Accra, Global Excelsior. We have a small booth back there. Please come and have a look. We don't have free coffee for you this time, but the next time we'll arrange one. but definitely, these guys are doing a great job.
Andy, we have 2 minutes.
Thank you very much. So, as we mentioned yesterday, we have the exhibition stands there, both for the scientific and laboratory companies, and also for the Ghanaian artifacts. Please make some time to visit them. They are just behind there. They have beautiful Ghanaian dresses and a lot of goodies you can take with you when you return. So we are going to have a very quick coffee break this right after my announcement, and then we'll be back in 15 minutes. Thank you.
10:40. Thank you. Come back at 10:40.
spoke. I did it yesterday, and I thought I had sent it. I just realized, so. Yes, yes. So it's without it. Yeah. 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 So there are two different files. Yes, yes. The moment you sent the reminder WhatsApp, yeah, because I did it yesterday and I thought I sent it. So the moment you sent it, I was like, oh, what? So immediately I just sent it. Did everything online come? You know it's online. You want to see it here? No, you have to see it here. And so you will check it and tell me because it's streaming live. So we have to. You see, they don't use coupons for hello. No, I meant that when okay, whatever is projected is streaming live, which is still combining with videos. So
not just you put out anything as you do something else. Yeah, that's right. It incorporates into the whole thing. So it's not like, oh, we'll break this thing, put it on the screen for us, whilst we do our thing. Because whatever you're doing is not for people here. So how does what you're putting on the screen incorporate into what happens there when you do that? You can't see from.
So, okay. So is the program outlined for that particular period? So during the pitch competition?
Yes, so the program outline, is it for people online throughout the time, or is just brief display and it goes off so that the pitch itself would be?
No, so for that, let's say 1 hour and 25 minutes. I guess it's structured. You know, at this time.
No, what I'm saying is, yeah, the program, everybody knows, it's like the program for this thing. We don't put it out.
No, that's not it. Like
We don't put it on this.
No, I'm saying here the program was the projections. That's what I'm referring to. So for
Okay,
example,
so let's just do this. Exactly, sure. So you give it to us for our perusal,
Yes,
not
that's
for
what
us to put it on the screen.
Okay, so for instance, we have.
Based on the conversation you said first, it sounded like you want to give it to us to put it on the screen for you because you said we are judging. Yes. Are we putting the program on the screen?
No, not the program. I said the way you have
Exactly. So it's rather more important to collate all the presentations and just number them, which one is first, second, third.
That's the reason to we are not predicting it. But I would just have to work with them.
Yes, the reason I'm saying that is usually in the layout of the program. You see the way like all the cars are just a little bit on top of this. That's all I'm doing. Police there. I said this is the second.
one do you want? For Illuminous or for me? Okay. Okay, should I send it to you on WhatsApp? Okay.
Hello there.
Get rid of the hassle of procuring lab supplies and start running and continue. Hello there, we're Excelsior Global Limited. We are back with exciting procurement and supply solutions for you. Take a moment to imagine a laboratory where procurement was such an easy task. Our lab equipment and consumables are only of the highest quality, and Excelsior procures these from the most highly regarded manufacturers. Our lab supplies include equipment, reagents, and consumables that you use every day in your lab. Get rid of the hassle of procuring lab supplies and start running your laboratory smoothly. Take your lab experience to the next level with our game-changing solutions. Choose Excelsior. Hello there, we're Excelsior Global Limited. We are back with exciting procurement and supply solutions for you. Take a moment to imagine a laboratory where procurement was such an easy task. Our lab equipment and consumables are only of the highest quality, and Excelsior procures these from the most highly regarded manufacturers. Our lab supplies include equipment, reagents, and consumables that you use every day in your lab. Get rid of the hassle of procuring lab supplies and start running your laboratory smoothly. Take your lab experience to the next level with our game-changing solutions. Choose Excelsior. Hello there, we're Excelsior Global Limited. We are back with exciting procurement and supply solutions for you. Take a moment to imagine a laboratory where procurement was such an easy task. Our lab equipment and consumables are only of the highest quality, and Excelsior procures these from the most highly regarded manufacturers. Our lab supplies include equipment, reagents, and consumables that you use every day in your lab. Get rid of the hassle of procuring lab supplies and start running your laboratory smoothly. Take your lab experience to the next level with our game-changing solutions. Choose Excelsior. Hello there, we're Excelsior Global Limited. We are back with exciting
What's your name? No, I mean, it doesn't matter. It's not enough proof of security access. She just so if she wanted a key card, if it was a guy, you would have failed. Because it's a lady. But she came for it earlier on. I don't know. She came for the key earlier on. I think so.
Hello there, we're Excelsior Global Limited, and we are back with exciting procurement and supply solutions for you. Take a moment to imagine a laboratory where procurement was such an easy task. Our lab equipment and consumables are only of the highest quality, and Excelsior procures these from the most highly regarded manufacturers. Our lab supplies include equipment, reagents, and consumables that you use every day in your lab. Get rid of the hassle of procuring lab supplies and start running your laboratory smoothly. Take your lab experience to the next level with our game-changing solutions. Choose Excelsior.
WACCBIP came out of the energy of many young people in the department at the time.
WACCBIP's excellence really stems from teamwork.
We are at the cutting edge of technology.
We make excellence the bar for training at WACCBIP.
We believe in excellence in teaching and excellence in research.
To do high quality science, to be able to publish and to make an influence in the scientific world.
The West African Center for Cell Biology of Infectious Pathogens, WACCBIP, began in 2014 as one of the World Bank African Centers of Excellence, located in the University of Ghana, Legon. In 10 years, it has grown into a leading center for the training of the next generation of African scientists and is spearheading groundbreaking research into infectious diseases, genetics research, and research into non-communicable diseases like cancer. Equipped with advanced technology platforms and state-of-the-art laboratories, WACCBIP provides fertile ground for exploration and discovery.
The main impetus for the center came from some workshops that we used to do with the American Society for Cell Biology.
It was my honor and pleasure to be part of a group of cell biologists who obtained funding around 2006 for short courses on the cell biology of African diseases.
This was long before WACCBIP was even a dream, and the workshop was hosted at the old Department of Biochemistry, which exists down the road from where WACCBIP currently stands.
In 2010, with our move to this building, we had space. And also, that also coincided with a period that we had younger faculty who had undergone training in the department.
It was around that time that I had come back to the department. And the head of department at the time, who was Prof. Sami Saki, asked me to be responsible for these workshops, to be sort of the coordinator for these workshops. We were able to engage with a lot of young people in the sub-region because each workshop we had about 20 to 25 young scientists from from Ghana and other African countries, and the conversations we had, and their desire to learn, and the appetite for such opportunities. That was what drove us to think about how we could make this a permanent presence or a permanent opportunity for young African scientists.
I wrote a grant application designed to obtain more funds to run more of these workshops. My application was not successful, but Gordon, whose vision is much broader than mine, was able to take some of the ideas that we had put into that application and expand them tremendously and significantly into an application that ultimately was submitted to the World Bank, and many of you know was successful.
The idea about WACCBIP really was a call from the World Bank trying to help higher institutions to set up the BIP. So I called Gordon and said, Gordon, I think you should host this.
So basically, I came back to the department and I got all my colleagues together, mostly the younger ones who were full of energy and who listened to me because they were younger than me. So I put them together, you know, Patrick, Lydia, Theresa, Osborne, Abida. There was another lady called Marian Yako. The vice chancellor at the time, Professor Ernest Aite, was determined to turn the university into a research-intensive university.
WACCBIP first came to my attention when Professor Gordon Wandari approached me about the possibility of creating such a center. And I thought they were very important for the university. I was struck by the fact that he himself was a very young researcher. Most of the people involved with him were equally young. And I was looking for young people at the university to drive change.
An initial $8 million grant from the World Bank and further funds from the Wellcome Trust allowed WACCBIP to build a world-class center adjoining the Department of Biochemistry. Equipped with teaching labs and creating an environment that fosters scientific research and learning. The center runs master's, PhD, and postdoctoral training.
I'm very proud of WACCBIP because it has leveraged on the initial funding of $8 million from the World Bank and attracted more than 50 million US dollars for training of scientists right here in Africa.
WACCBIP's core mandate was around infectious diseases. Now, as we grew over the years, we noticed that non-communicable diseases are also coming up strongly in our part of the world. And so our funding portfolio evolved to starting to apply for grants that were involved in non-communicable diseases. Our ability to evolve with changing research interests over the years, I think, has kept us relevant.
It's been an honor for me and a privilege to help in some small way with that early development, and then to chair WACCBIP's advisory board over these last 10 years. A key success of that teaching of WACCBIP in the early years was the contribution of early career faculty, both from the North and also local faculty from Africa.
The next phase of the center was to consolidate what we had started and then to basically make an impact.
So began an investment into recruiting some of the best African scientific minds to join the center as postdoctoral fellows to conduct research and to teach.
I was looking for opportunities to come back, and because I wanted to drive research on the continent.
I was one of many immunologists at my institution in London. I'm one of only a few immunologists in Ghana. So when I think of where I can have impact, if I look back at my life when I retire one day, where can I have the greatest impact? It is in trying to build up the ecosystem back home where we are admittedly, a little bit deficient, versus making a small contribution to an ecosystem that's already well established.
As a geneticist, I was keenly aware of the depth of genetics research in African
some
people.
Fruit,
and what was striking is
coffee.
It's a huge,
especially coffee, so that we're energized for the rest of the day. we have a few changes to the schedule. So we're going to skip the plenary 6 talk, but before that, I'll call, so we'll go straight to the fellow session. So I'll call the 2 chairs who are going to chair the fellow session. Dr. Irene Owusu-Donko from Noguchi, who is also one of my favorite co-supervisors. And then Dr. Abiola Isawumi, who is also one of our fellows and a WACCBIP alumnus.
Good morning, everybody. So we'll go straight to the first fellow session. We'll invite Philip Lani, I hope I pronounced his name right, to give us his talk.
Okay, good day, everyone. My name is Philip Ilani. I'm a third-year PhD fellow here at WACCBIP, and I'm happy to share with you today some of the work I've been doing on functionally characterizing myosin motor proteins in Plasmodium falciparum. I'd like to start off this way. If I was a parasite and I was given these 2 options, to live in an island, a villa with all the luxurious things you can think of, or to live in a desert, I'd definitely go with option 1, but not Plasmodium falciparum. This parasite has chosen to reside in cells that are more or less referred to as molecular deserts. And that's because these cells lack the established protein trafficking mechanisms that are present in these other cells. And so the parasite invade red blood cells, renovate the cells to suit their purpose. And it's quite fascinating, and I think it's important for us to study the biology of this parasite. And so the problem is these parasites are developing resistance to the frontline antimalarials that we have, and developing vaccine has been a challenge over the years. And so we need to develop more arsenals to tackle malaria. If you look at the life cycle of the parasite, ranging from when the mosquito bites, every single stage requires the activity of certain key molecules. And the parasite needs this to generate force, to move molecules around in the cell, and to maintain shape, amongst others. And these stage-specific events require key proteins like the myosin motor proteins. And so looking at the erythrocyte invasion as an example, we see one of these myosin motor proteins, myosin A, providing the force necessary to penetrate the RBC. What are these myosin motor proteins? These are mechanoenzymes that hydrolyze ATP, releasing inorganic phosphate, and this way the protein is able to undergo a conformational change, and this way is able to walk along actin tracks. This process is known as the ATPase cycle, and this cycle is important for the function of the protein. Interestingly, Plasmodium myosins are quite divergent from human myosins, and that is why it gives us the avenue to be able to develop therapeutics using this approach. Interestingly, myosin A, which is one of the myosins I mentioned earlier, there's been a recent publication where a small molecule inhibitor has been identified to bind to this myosin, affecting its ATPase activity. This small molecule inhibitor is able to kill parasites in the nanomolar range. And interestingly, it can, this molecule can even kill parasites that are already resistant to the current antimalarials that we currently use. And this compound has also been shown to target several stages of the parasite development, which are desirable properties of a drug, I would say. But that's not the only myosin we have in Plasmodium. There are 5 others, but we know so much about myosin A and know very little about these ones. If you compare these other myosins, they have long tails and they have interesting domains that we really do not know what they do in the parasite, and that's been the focus of my PhD, trying to understand what these myosins are and what they do in the parasite. A paper from Rita Tewari's group tried to knock out these myosins in Plasmodium berghei, and they found that when they knock out myosin A, F, and K, it blocks the asexual development of parasites, indicating that they could be essential. And so I quickly looked at the transcriptome data available online and tried to look at the transcript abundance of these myosins. And interestingly, I found that myosin J, for instance, has an upregulated transcript abundance towards the late stage of the parasite development. And so I focused on myosin J, even though I've alongside been working on myosin F. And so the question we started asking is, are these myosins essential for parasite growth and development in Plasmodium falciparum? And to answer this question, I used CRISPR-Cas mediated approach to generate transgenic parasites where we have added a ribozyme to the 3' end of myosin J, along with a triple HA tag. And what happens is this, once the mRNA is transcribed in these transgenic parasites, this Glimix ribozyme forms secondary hairpins, and upon addition of glucosamine to your culture media, glucosamine binds to the Glimix sequence and then leads to the degradation of the mRNA. And this way we can downregulate the protein that is produced in the process. And so I cloned this construct into the respective plasmids, sequenced them, and once I was happy with the sequences, I went on to do transfection. When I got transfectants back, it was important for me to test whether these transfectants are really transgenic parasites or I've got wild-type parasites back. And so I use primers that are specific to wild-type and primers that can help me differentiate between wild-type and the modified locus, which I have listed in the key here. And so when I did the genotyping PCR, I was able to amplify the sequences for the wild-type, as you see in 3D7 here. And then I got successful transfectants back, although you see that I still get some wild type mixed. And so I went on to do a plaque assay to be able to select single clones of the parasite, which I then used on for the subsequent experiment. And so the next question I asked, because our protein is tagged with HA tag, I sought to test whether anti-HA antibody could detect the endogenous parasites are the correct ones. And so I started asking the question where myosin J is localized in the asexual stage parasites. And so I did immunofluorescent assays where I labeled parasites with both pre-immune and anti-MyoJ antibody, which I'll talk about shortly. We generated antibodies against the tail fragments of recombinantly expressed myosin J tail. Where you seed parasites on cover slips and then embed them in a gel, and literally you can expand the gel. Because we know the diameter of the gel we are starting with, you can actually know to what extent you've expanded the parasites. And when I did this and stained with the HA antibody, I was able to see a ring-like staining pattern in dividing schizons. And that gave us a clearer view compared to what we had for the IFAs. I just blew this up so you can appreciate it better. Although my MyoJ antibody could not give me a consistent staining, which is something I intend to go back to look at. While I was doing this paper was published, and they were able to detect myosin J in between 46 to 48 hours post-invasion at these segmenting schizons. And that kind of supports our data and even corresponds to the transcriptome data that I mentioned earlier. And so the question I then started asking is, if we remove myosin J, knock myosin J down, what happens to these segmenting schizons? Are we able to, and how would these parasites fare in growth assays? And so I induced the knockdown of myosin J by treating parasites with glucosamine, and I did not see any visible phenotype when I knocked down myosin J. I tried to hypothesize that although for some of these parasites, I saw some fuzzy-looking schizons, about 20 to 25% of them, and then I started asking the question, what is the viability of merozoites that would come from these schizons, if there's indeed an impact on segmentation? And clearly, I was unable to see any significant difference between treated and untreated when I did flow cytometry. And then I did Western blot just to show that myosin J, upon addition of glucosamine, you see the protein is knocked down. I quickly looked at the IMC to see if this would have any impact on proteins in the IMC like GAP45 and MTIP, and clearly I could not see any effect. Whether this phenotype will be the same in sexual stage parasites is something I'm currently doing in the lab. And the other part of my project is trying to look at the properties of this myosin, which I mentioned earlier. So I made this different construct, and I'm trying to produce the protein in baculoviral expression system. And these I will use for structural analysis and then try to identify the light chains, because myosins need light chains for their activity, and then also do pull downs to identify binding partners. Successfully, I've been able to make this recombinant protein in the insect cell expression system, different fragments of it, the motor neck, the tail, and the two tails. This is the tail we use to generate the antibodies. And because these proteins are tagged with GST, when I'm going to do pull down, I would also use GST as a control. So I've been able to also make recombinant GST. Quickly, I tested if anti-His and anti-GST antibodies can detect the recombinant protein. And clearly, we see the detection here in Western blot. And even myosin J antibody, tail antibody, which I talked about earlier, can also detect the recombinant protein compared to the pre-immune, where we see nothing. To sum it up, I've been able to show you that we can generate transgenic parasites for inducible knockdown of myosin. I've also been able to show you that myosin localizes to basal ring-like compartment in dividing schizons. Upon knockdown of myosin J, we really do not see any visible phenotype in asexual stage parasites, although I'm currently looking at sexual stage parasites to see if there will be any phenotype. And myosin J deficient parasites also showed no defect in asexual growth. And I've also shown you that we can produce the recombinant proteins now, which I would use for structural and biophysical characterization. I'd like to thank all the labs that have been involved in this work. I couldn't have been able to do this without the support of these people and these really amazing mentors and supervisors for their support. I'm grateful to WACCBIP, and on this 10th anniversary, I say congratulations to WACCBIP. Thank you all for listening.
Thank you, Philip. We'll make room for 2 questions.
Well done, Philip. That was beautiful.
Thank you.
So when you knocked down MyoJ, did you check to see if any of the other myosins were upregulated, or if there was any kind of, or are you considering doing double knockouts?
That's an interesting question. I haven't done that, but yes, I intend to do that, looking at the expression of these other myosins. I would be surprised if they would have any effect, except they are maybe working together or something. But yeah, it's something I definitely would want to check.
The other thing I was curious about is how long did you do your growth assay to look for a growth defect?
That's a really good question. I did this for 48 hours. I intend to repeat these growth assays for over 4 cycles. Maybe I can sufficiently knock down myosin J over a longer cycle than just the initial one I've done. So I've done this for just 48-hour life cycle. Yeah. Divergence between humans and Plasmodium myosins. I find these proteins very interesting because, to me, they seem to be all over the place in the parasites, and there are about 6 of them. We know so much about just one. And so I decided to look at the others, what they do. Interestingly, back in 2022, 2023, the paper on myosin A came out where they identified an inhibitor, KNX002, which binds to myosin A, reduces its ATPase activity, can kill parasites at different stages of parasite development, can even kill parasites that are resistant to the frontline antimalarials that we have. So I thought it would be an interesting shot to look at the other mycins. And mycin A, the inhibitor that's targeting that mycin, there's a whole company in the US now.
Hello, Eunice, can you hear me? Eunice, if you can hear me, can you put your presentation in, your slide in the presentation mode so that we can start the presentation? Please, technical group, can you hear Eunice? Can you confirm if she can hear me?
Yes, I can hear you.
Okay, please go ahead with your presentation. You have 12 minutes.
Thank you very much. Good afternoon, everyone. My name is Eunice Ampezia Iraq. I'm a second-year PhD fellow, and I'm going to give a talk on the phenotypic island and genomic structure of Klebsiella obtusa indicated in nosocomial infections in Ghana. So Klebsiella species are important human pathogens that form part of the normal here we have grouped the things that, or the markets we identified from the sequencing according to the mechanisms that we are indicated. And we compared the number of genes we had from our isolates to the ACEs control. And we found out that our isolates have acquired more resistance markers as compared to the ACEs control. And for some resistance mechanisms, there were no markers in the ACEs control, but we had some in our isolate. So the third aim was to confirm the violence of isolate using in vitro and in vivo assays. So for the in vitro assays, we used human macrophage cell lines. We incubated the encephalomine CSU of our isolate with the macrophage cell lines for 24 hours, and then we determined the viability of the cells after the 24 hours. So we found out that for both our isolates, cell viability was reduced to 80 and less than 80% compared to the ACEs control, which was reduced viability to about 60%. So we also determined the viable bacteria we have, which is an indication of how our pathogen is able to survive in the macrophage cell line with and then without antibiotics. So without antibiotics, you can see that after 48 hours, we have 100% survival of bacteria. And then in the presence of 10-24 microgram of DNA, this survival rate decreased a bit, decreased after the 48 hours. But then when we compared the ACEs control with our strain, we found out that our strain was a bit resistant, but after 48 hours, we can still have viable cells in there. So finally, we did an in vivo infection model with Gardnerella species. So we infect the Gardnerella species with 10 cells per larvae, and then incubate for 72 hours, and then determine the larvae survival. So again, here we realized that after 48 hours, we have complete healing for all larvae. And we know that one of the problem aspects of gum-related bacteria is the LPS. So we purified those LPS from our isolate, our theological isolate, and then infected the larvae with this whole process. And we also had similar observation that so we had complete scanning after 72 hours of infection compared to the ACEs control, which we observed no So in summary, our first aim tells us that Klebsiella obtusa is a strong biofilm former with high level of resistance to conventional and natural antibiotics. We also identified that the isolate has a tendency to elevate resistance and then antibiotic resistance. Our second aim also shows that the core did not contain markers of bacterial toxin export and then antibiotic resistance, which indicates the intrinsic ability of these pathogens to evade antibiotics. We also showed in our final aim that Klebsiella obtusa isolates are able to evade macrophage killing and could only 10 cells per larvae were enough to kill the Gardnerella species. So at the end of this study, the study tells us that Klebsiella obtusa is really emerging as a major public health threat in our hospital environment. And the fact that the isolate show resistance and persistent phenotype is as it is AMR contributes a contribution to antibiotic treatment failure. And then these findings also suggest that the isolate has genomic signatures that will drive a resistance profile and the virus profile we are observing in the first and the second objective. And these signatures will serve as the basis for developing targeted intervention. I would like to acknowledge my supervisors such as President Kamu, Dr. Andy, and Dr. Miguel Valvan. I would like to acknowledge WACCBIP and the Biotechnology and Research Council for the funding for this work. Thank you.
All right, thank you, Eunice, for the excellent talk. We have time for 2 questions. Okay.
Thank you very much for a very interesting talk. I wanted to know about the larva in vivo infection model, and if it's relevant only for Klebsiella or other pathogens as well.
You can use it for other species, not just Klebsiella.
Interesting, thank you.
So do we have any other question?
Thank you. Thanks for a very interesting, very nice talk. Could you tell us a bit more about the epidemiology or the public health perspective on this pathogen? Is it a widespread problem in different hospitals? Yes, thank you. Yes, please.
All right, thank you, Eunice, for the talk.
So we'll take the final talk from Harry Hayford.
Hello. Can you hear me? Okay, good morning. My name is Harry Hayford, and I'll be presenting on the role of histamine production in Klebsiella pneumonia antimicrobial resistance and virulence. So histamine, as you already know, plays major important roles in the human body in neurotransmission and pro-inflammatory responses. However, exogenous histamine from other sources outside of the human body have been found to cause allergic reactions and cause histamine intolerance. The major sources of these exogenous histamine is as a result of the decarboxylation of the amino acid histidine to produce histamine in bacteria. These bacteria produce histamine in order to survive extreme acidic pH so that they can survive in these acidic pHs. The most common bacteria, or one of the most common bacteria that produce histamine in large quantities is Klebsiella pneumonia. The bacterium is a gram-negative bacterium that has been characterized as a global priority pathogen by the World Health Organization and may cause different complications based on the area of localization in the body. It is currently treated with cephalosporins, quinolones, and carbapenems. However, resistance to these antibiotics, as well as other classes of antibiotics, is a common occurrence in Klebsiella pneumonia, which is thought to be spread through the presence of high-rate clones and resistant plasmids. In addition to this, the bacterium can produce biofilms which enable them to survive in the environment to cause nosocomial infections. And also, they are able to produce capsules which ensure their survival against phagocytosis. So although histamine production has been characterized as a survival mechanism to environmental stress like acidic pH, the effect of histamine production on other survival mechanisms like antimicrobial resistance and biofilm formation is not known. So this study sought to investigate histamine production in Klebsiella pneumonia by detecting and quantifying histamine production in vitro, determining the effect of histamine production on antimicrobial resistance and virulence, and also determining the effect of histamine production on the expression of antimicrobial resistance and virulence genes in Klebsiella pneumonia. So the study employed 4 strains which were archived. 2 were clinical from septicemia patients and 2 were environmental from hospital formites. For the histamine detection assays, we use E. coli EK19 as a control. So the experimental approach was basically first to detect histamine, quantify it using enzymatic methods in HPLC. Next, we did comparative antibiotic susceptibility testing by determining or testing the resistance and susceptibility of the histamine producing and non-histamine producing strains of Klebsiella pneumonia. Then we went ahead to do infection assays to determine the effect of histamine production on virulence in Klebsiella pneumonia, and also expression profiling using RT-qPCR to determine the effect of histamine production on the expression of resistance and virulence genes. So to begin with, we found that the strains could produce histamine. And this assay is the Nevin's calorimetric assay. The change in coloration of the media from the brick red coloration to the purple coloration, as can be seen on the screen, is as a result of histamine being produced. So the media is acidic. Once histamine is produced, it changes the coloration of the media to purple, which is as a result of the increase in the pH of the media. We determined that histamine production in all the strains began at the log phase. We did this by isolating cells from both the log and stationary phases and testing them for histamine production. In this case, we found that since we found that histamine production was produced at the log phase or began at the log phase, it made us hypothesize that because it is required to survive the extreme acidic environment, then there is the need for the strains to produce histamine even before the log phase in order to undergo exponential growth. Next, we used HPLC to confirm and quantify the amount of histamine produced. In all the tests for the strains, we found that we detected histamine at a retention time of 26.5 minutes. And also, the strains could produce histamine between 390 to 800 parts per million. In all strains, we found more histamine was produced at the stationary phase as compared to the log phase. This could be indicating that histamine could be advantageous in the stationary phase in order to survive the increased bacterial competition, which might be due to the production of antimicrobial secondary metabolites. Next, we screened the strains for the presence of the histidine decarboxylase gene, which is responsible for histamine production. We found that in all the strains, the gene was present. So going forward, we wanted to determine the effect of histamine production on antimicrobial resistance and virulence. So here, what we did was we induced histamine production in the strains and then just did antibiotic susceptibility testing and all the other tests for the phenotypes. So after ASTs, we found that the resistance profiles of the strains did not change after histamine production, indicating that the strains were still resistant to or susceptible to the antibiotics they were resistant to before histamine production. However, we found that there was increased antimicrobial resistance levels after histamine production. So if you look on the screen, the previous MICs, that is the minimum inventory concentrations that could reduce the bacterial growth to less than 10% before histamine production, were no longer effective in reducing the bacterial growth to less than 10% after histamine production, as is shown on the screen. And after doubling the concentrations, that is the minimum inventory concentrations, we found that still they were ineffective in reducing the bacterial growth to less than 10%. In the case of the antibiotic meropenem, we found that after histamine production, when you increase the drug pressure by doubling the minimum inventory concentration, there's actually an increase in survival, meaning that there's increase in resistance after histamine production. Next, we determined the effect on biofilm formation. So here we observed two things. In the case of strains that produce high concentrations of histamine, here, you found that after histamine production, there is reduced biofilm formation. But in the case of strains that produce lower concentrations of histamine, after histamine production, there is increased biofilm formation. So this is indicating that histamine production is reducing the need for biofilms to be produced. So biofilms are also produced in bacteria to also help them to survive the acidic environment. So in the case where there's more histamine being produced, there's lower acidity in the environment. That means the environment is more favorable for the bacterium to survive. So it requires it to produce less biofilms in order for it to survive. Next, we determined the effect of histamine production on encapsulation. We found that after histamine production, there were more encapsulated cells as compared to before histamine production. This could be indicating that histamine production could be driving the cells to survive phagocytosis. So to investigate this, we infected human macrophage cell lines with both histamine-producing and non-histamine-producing variants of Klebsiella pneumoniae. And then we found that histamine production did not cause a difference or a change in the viability of the macrophage cell lines. That is, they still had the same degree of cytocidal effect on the cell lines before and after histamine production. However, we found that after histamine production, more cells could survive the macrophage phagocytosis by the macrophage cell lines as compared to before histamine production. So this can be attributed to two things, may be attributed to two things. It's either from encapsulation of the increased encapsulation in the cane pneumonia strains, or it could be as a result of the effect of the histamine itself being produced on the macrophage cell lines. So next, we screen the strains for common resistance and virulence genes that are normally observed in Klebsiella pneumonia. We found that the strains harbored at least 2 resistant genes for beta-lactams and efflux pump genes. In the case of virulence genes, the strains harbored all the strains harbored virulence genes for fimbriae, siderophos, and membrane lipoproteins. So to determine the effect on gene expression, we use RT-qPCR. We found that in the case of the resistant genes, they were downregulated after histamine production, whilst in the case of virulence genes, the virulence genes were upregulated by histamine production. Now, the upregulation of the virulence genes was expected because there was increased encapsulation after histamine production, and also in the upregulation of the fimbriae genes could be indicating why biofilm formation was altered after histamine production. So, in summary, we found that histamine production increases antimicrobial resistance levels in Klebsiella pneumonia. It complements biofilm formation for survival in an acidic environment, and it increases virulence by promoting survival in macrophages. And since the strains could produce such high concentrations of histamine, it indicates that they have the possibility to cause histamine intolerance in addition to all the complications they might cause. So in the presence of these evidence, we could say that histamine production is a significant public health threat that should be investigated further. I would like to acknowledge my supervisors, my labs, and the Ghana Standards Authority for the study, and also WACCBIP for funding. Thank you.
Thank you, Harry. We'll take 2 questions.
Those who are more susceptible to allergens
yes,
are more susceptible to infectious diseases. Yes. Okay, so the studies have shown that for some kinds of diseases, so for those who are more susceptible to allergens, they have larger populations or higher populations of bacteria that harbor the HDC gene. So in the case of asthmatics, and also people with irritable bowel syndrome, studies have shown that they have larger populations of these bacteria in there. But as to whether they are more susceptible to all infectious diseases, I can't really talk about that.
Thank you. I just have, I think it's more of clarification for me. When you did you said histamine increases the phagocytosis of the Klebsiella in macrophages.
It promoted their survival in the macrophage cells.
So they make the cells survive?
Yes.
And then so the cells survive, the parasite survives?
Yes.
The bacteria survive. Okay, thank you.
No more questions. All right, thank you very much, Harry.
All right, thank you, Harry. There is a slight adjustment in our program. Earlier today, we missed a plenary talk by our very dear own Dr. Yao Aniwe. So we'll be having Dr. Yao now. He's a senior research fellow here at WACCBIP, at the same time, the head of technology. Dr. Yao Aniwe, you are welcome for your plenary talk, please.
No problem.
Okay, thank you very much for the opportunity to present what we've been trying to do. Because I've already taken your time, I will not spend much time. I will present what. So, looking at malaria, there are a number of parasites that are able to cause malaria. Like the presentations that have come before, you will hear Falciparum as the major. But notwithstanding, there are other parasites that are also driving malaria in our settings. So in Ghana, we have malaria in ovale as one of the major. And you will see most of the time, anytime you have Falciparum, the major culprit who is underlining also, or going where it is Plasmodium malariae and Plasmodium ovale. Most of these are known to cause benign disease. That is what we have known them for. Others, chronic infections of P. malariae has been associated with nephrotic syndrome. Other complications have been shown. Recently, there are a number of reports of anemia associated with monoinfections of these parasites. So the simple question anyone will ask is that, really, is that the case? If you look into our population, if chronic infections of P. malariae can drive nephrotic syndrome, then could it be that a number of people who are in the kidney clinics, we should begin to ask some of these questions in there? So what we have been trying to do is to help us be able to, first of all, detect them, and then we can now begin to look into different populations. One thing which is very common with these parasites is that they always occur in low parasitemia, and most of the time occur in co-infections. One thing is the diagnostic tools we have. Whether microscopy, you need a trained microscopist who is diligent enough to find these. Partly also, the RDTs we have don't pick. Most of them are HRP2 based, and therefore wouldn't be able to pick this except pan-LDH. So that is also another question. Since the pan-RDTs are more expensive, hospitals don't use them mostly. So if you look through literature, you'll find that there's not much done on these other parasites, partly because we consider them neglected. So recently there have been reports that after treatment with ACTs, these parasites persist. So the simple question that everyone will ask is that, do the ACTs actually have direct impact on non-falciparum malaria? And recently, with growing data, clearly we are seeing that it's not that effective against the non-falciparum malaria. So if you to look at falciparum in the blood, you will always find the ring stages. But if you get to P. malariae and P. ovale, you could find all the different stages, including gametocyte in the blood. So this is one of the unique challenges also with them. With P. malariae, the life cycle is a 72-hour life cycle, while these are 48-hour life cycles. P. malariae is known that it is able to form a hypnozoid, meaning that it forms a dormant form which hides in the liver, and whenever they are happy, they will come out. So if you give somebody a drug and then the drug pressure wanes, they decide to stay or decide to come out. So these are the conundrum. But notwithstanding, recently there have not been any data really pointing to the hypnozoid forming ability of P. ovale, which is still something to be explored in the community. So one particular thing in the lab, we developed tools, courtesy talented PhD students and graduate students. So these allow us to be able to now detect the non-falciparum which occur in low parasitemias, now with good confidence, and then we could go and classify them. So one particular thing is that clearly, if you look in this, what you see is that Falciparum, this is a clinical case. We always have falciparum being the dominant, which is what we know. But notwithstanding, you could have up to 20% of the people going to the hospital being infected with malaria as well as ovale. Then when you want to segregate this and ask the simple question, do they occur as monoinfections or mixed infections? You'll find still you have monoinfections of falciparum dominating, but you have mixed infections of falciparum and malaria being the second, and then you have monoinfection. And in some individuals, we could have somebody having all the three jubilating in the person. Yeah. So in terms of age distribution, this one, we need not belabor it. It occurs, it's very common in the young, so you can see that. But notwithstanding, still, in these individuals, you will find malaria. With the age group distribution, you find the same pattern. So clearly, here is just to show you that if you use microscopy and then the qPCR, we know qPCR is at the molecular level and therefore it really helps. With falciparum, because we always have higher parasite numbers, it is easy to find them. But with that will have broad spectrum, and as you will have listened to Franklin's talk yesterday, we're trying to look at the CSP, which is cross-reactive to falciparum, ovale, and malaria. Yeah. So here specifically also you will see that those who have mixed infections, yeah, there are not too many, but actually they are there. And we have to be concerned and to see how interventions targeting these individuals will help us drive the burden of the disease down. Yeah. So one particular thing that we try to do is we have these parasites. They are co-occurring. We have fewer monoinfections. The simple question is that, can we now develop tools to help us be able to study their drug susceptibility pattern? So what we did was to get monoinfections of P. ovale and P. malaria. And the other criteria is to be able to see individuals who have relatively higher parasitemia, and the composition of the different stages of the parasite should be biased towards ring stages so that you can now follow them through. So with this, we developed the protocol for P. malaria. And here I show you susceptibility. We compared the protocol between Ghana and Mali. So here, using chloroquine, you will realize that, yes, most of the parasites in the population have become susceptible to chloroquine, but with P. malaria, you still get a few that shows kind of resistance. Comparing it to quinine, others, if you look at Artemita, there is this parasite that decided not to really succumb to. One particular thing is that there is the likelihood that some of these parasites are more of the late stages. But if it is the late stages also, we expect these drugs, which we know kill trophozoite and Schizon stages very well, to be very effective in killing. But then, irrespective of whatever stage, we still get some of the parasite that shows a higher level of tolerance to the drug that is used here. So the other thing is that we did this same study also. The earlier data is Mali, here is for Ghana. We find some malaria that is also. So this is actually the simple question we're asking. Are these parasites resistant? And if they are, what are the unique markers? As we can say now, most of the resistance we know, and the associated genes are all for falciparum. We don't know for malaria and ovale. So this is one of the things that we're doing to now begin to go down and ask these questions. What genes are driving this phenotype?
A very beautiful talk, by the way. We'll take questions, but I would like to start. So, was there a reason why you picked Mali Was there any specific reason comparing Ghana and Mali?
No, we could have used any other, but I think with Ghana, Mali, I have an active collaboration there, and this protocol was developed in such a way that both labs could test. So now we're bringing in Benin and other countries to equally adapt to that.
All right, thank you. Okay, we have a question at the back. So there's a question.
Can I?
Go ahead.
Yes. Go ahead.
Okay,
please.
thank you very much. Thank you anyway to present this. Okay, I'm very concerned about what you've said. You said Plasmodium ovale and malaria are really very low in terms of prevalence. But at the same time, it can happen in symptomatic and asymptomatic. So my question is, did you actually test this in hospitals? And in that case, don't you think there has been a selection bias and showing that this prevalence might be high, much more higher than what we think?
Yeah. Actually, we actively telling people to be testing for this simply because, one, there is a hypothesis that when you treat, because they are less susceptible to the ACTs, they will be amplified. So there's a recent paper from Tanzania showing a community that to them, they've driven falciparum down and they are seeing malaria come up. So that is one hypothesis. The other thing is what we call the comparative inhibition. In the presence of falciparum, the others are quiet. And as long as falciparum decide to go with any drug or any intervention, then they now get up to jubilate in the house. So these are some of the things. There are not many people testing. Because most of the testing we do are PCR based. That cannot be done in the hospital. But gradually, with tools like LAMP, we are getting the hospitals to also begin to test this. Yep.
Okay.
Yes, thanks, Yael, for that nice presentation. Just to check, for the complexity of infection, because you said you detected more monoclonal infections, but I think in some other publications, including ours and yours, there seems to be quite a lot of diversity which is not geographically restricted in P. malaria, for example. So it was quite surprising that having that level of complexity population-wise, single infections were always monoclonal. Do you have any explanation for that? The second is the work that you're doing across laboratories. For P. falciparum, we found out that its variability between laboratories, even though you're sharing protocol, can be a significant nightmare, a significant nuance to the data, and to be able to compare data between labs. So what are you using to normalize the phenotypes you're getting, IC50s in this case, between the two labs? Because it will become quite difficult to compare if you don't have a comparator as a control that you can reference to.
I think the last part, I don't know, did you guys hear?
Yeah, the last one was how do you compare? Because you showed those dot plots comparing Mali and Ghana.
Yes.
And my problem there, as for P. falciparum, is that you cannot really compare assays between two labs because interlab variability is quite high. Even within the same lab, two individuals running two assays for the same isolate for IC50 may end up giving you different results in terms of numbers. Yeah. So the distribution of those numbers compared between two labs, how do you control and normalize between the two?
Yeah. Okay. So with that, what we did was we did the assay. We initially used falciparum. So the 3D7, DD2, and K1 are the parasite we use. We did the assay in Ghana. We have the IC50. But also with the Mali, I had to go there, help them set up so we could do same and make sure that the level of reproducibility in there is comparable. It wouldn't be the same, but it's comparable. Yeah. The other thing with the complexity of infection in terms of the different species, one thing I think is, So a work done by Felix in the lab, we took, say, Central Region specifically. We moved from town to town, and maybe town A, you'll find falciparum and malaria. You go to the nearby town, you don't find malaria, you rather find ovale. So the question has to also do with, is the vector competence the same? That is the other thing that we don't know. And I know there are groups who are looking into that. The other thing is also, in terms of the multiplicity of infection having the different species, what I know, because all these people or these individuals, we check their hematological indices, what is typical known to correlate with, say, like platelet count, the hemoglobin levels, are the same parameters that flag for all these as well. So, I mean, this is actually the reasons why we're amplifying this, given this talk is equally amplifying, to make sure that we all become active in looking for them and also looking towards finding interventions that will help us clear them. Yeah.
I think because of time, okay, we can have 2 more questions, then we can continue the conversation over lunch. So we can have, and then we'll come here, please.
Okay, so with respect to the mixed species infections, did you look for gametocytes so we can understand the level of competition between the species to get transmitted to the mosquito stage? So if you have falciparum and malaria, which one is making more gametocytes?
Yeah. So if you look at individuals that have mixed infections, you will mostly find, especially malaria, you'll find a malaria gametocyte. But that, I wouldn't compare it immediately with falciparum of the same individual, because falciparum getting into gametocytogenesis is triggered by a lot of factors. But with malaria, we know that, I don't know the biology driving it, but at any particular point in time, you sample somebody who has malaria, you find ring, you find trough, you find the gametocytes. This is one thing, but I don't know whether is it that they cannot sequester, or possibly there are unique mechanism they feel, let everyone go out and play, and if one is killed, the others will survive. I mean, these are all hypotheses that are to be tested. Yeah.
Okay.
Thanks, Jiao. So many years ago, when there was high falciparum transmission on the coast of Kenya, we looked at barley phenotypically, not using PCR, and found that although there were very low apparent prevalence, in fact, there was a very sharp age drop-off in the very young, implying that, in fact, probably it's being transmitted very widely, and then becoming immune quite quickly. I wonder whether you see the same pattern. And also, I think, to simplify it, you talked about Avali rather than the 2 species of Avali. Do you see any difference in distribution of Curtisii and Walkeri?
Yeah. So this is one thing that specifically I was about 4 or 5 cases were referred. Somebody who had been given antimalarials and still went back, and the person came when we checked. It was Ovale Walekeri. So kind of, in a way, we see more of Walekeri more associated with mono infections getting to hospital from our hands. This is what we're seeing. And also, Ketisi, you get them mostly with the data we have. All the samples we get from the community, the asymptomatics, you do find Ketisi as well more. Yeah.
All right, thank you, Dr. Aniwe, and again to all our presenters for this session.
All right, let's thank the chairs for a great job, well done. And as we say, time flies when you're having fun, right? It's almost time for lunch. But before we go for lunch, just a quick note about the poster sessions. So we will have Turbo Talks today, and the people who have been pre-selected to give Turbo Talks, the odd-numbered abstracts will give their Turbo Talks today in chronological sequence, so 1, 3, 5, and so on. So you have to make sure, even though we asked to submit the posters for us to print, those that didn't print, you have to make sure that you put up your posters if you're an odd-numbered poster, so that people can go around and look at it today and judge it during the poster sessions. So that's it. And for your poster numbers, look in the abstract booklet, which is online, if you haven't already done so. So without much ado, we're going to break. I think it's early. Yes. We're going to break early for lunch, which will give all of us a chance to walk around, look at the booths, speak to the vendors, look at the posters, speak to the presenters, and we'll be back here at 2 p.m. sharp for the keynotes from Professor Basiru Bonfo. Thanks for being sharp, and thanks to all the speakers for keeping within time.
Hello. Good afternoon. Welcome back from lunch. I hope everyone is awake. Otherwise, there's coffee outside for sale, which you can patronize. Okay, so this afternoon, I am truly honored to be introducing the second keynote speaker for today. Professor Basiru Bomfo trained as a veterinarian, but he also holds a PhD in epidemiology. He worked for a while with, I think, the Veterinarians Without Borders, and then worked as a postdoc fellow with the Swiss TPH for about 4 years. And then he moved back to Africa, and worked to establish what we know as the One Health concept right now. I can't even begin to tell you how instrumental he's been trying to bridge the gap between the North and the South, and also to foster South-South collaboration within Africa. What I'm most proud of this gentleman is he is the one who gave me my first break into life back after a PhD in the West. When I finished my training in Tennessee and I moved back to Ghana and was hungrily looking for a job, he interviewed me over Skype. I was about 7 months pregnant then, with my face all puffy. And I told him that, well, this is my situation, and he asked me whether pregnancy was a disease. I said, no, it's not a disease. As long as you be able to deliver on the job that you are applying for, by all means. And so it was part of the...
Merci beaucoup, Lydia. Très belle introduction. Je pensais que tu allais la faire en français. Comment est ton français? Comment est ton français? Okay, good afternoon. What Lydia just said, I could have just stopped my talk asking you some question around what we did. We just stand here and... So thank you very much. As she said, I'm from Togo, and I could have finished my career without knowing what is happening in Ghana. It's just 20 to 25-minute flight from Lome to Accra. But some of the initiatives gave me this chance to interact directly and indirectly with Ghana, and even beyond Ghana and Togo, from Cote d'Ivoire to East and West Southern Africa. So before I start my talk, I just want to recognize the power of the Deltas program. And I don't know if Kevin Marsh is in this room. Just want to acknowledge his contribution for the design of this program, and also thank Suzanne, she's there, who's really supporting us running the Deltas program. I don't know if Denise is around. Denise? Yeah, thank you very much. As she said, I'm from Centre Suisse de Recherche Scientifique in Côte d'Ivoire, which is a regional research center. With the name, you may think that it's Swiss, but it's an African institution. We just kept the name Swiss for the branding, because when you hear Swiss, then you don't even ask yourself, you can give money. And I'm the director of the consortium Afrique One, and Afrique One is one of the consortium like WACCBIP, and we run that with other 14 fellows. And the 14 fellows of Deltas, I think Gordon is here, they decided to put in my hand hot potatoes. Just to coordinate, I will not say the mess, but the most difficult part of coordinating the health research of 14 brilliant leaders, research leaders. And you have heard yesterday with what have been said about Gordon and his colleagues' work here. So this is what we call ASH, the African Research Consortium on Health Research. I don't know if I have. So what I'm going to talk this afternoon is just to give you an insight of what Deltas has done for the last 6 to, I will say, 10 years, and bring to your attention one of the concepts or the approach we have developed, which is One Health. And I will define that for you to understand. And since yesterday, I have been listening to most of the talk, and when I see what is happening in the lab with nice pictures from the lab, I just ask myself, what's the meaning for a farmer in a village? And what could be the impact of that for a farmer or a woman who is selling food at the market? I don't say that basic science is not important, it's very important, but sometimes we have to stop and see what are the pathways to the impact on the life of people. So everything started for me when I did my DV, and I said, so on which topic? And then I read the application, and they said, you choose the topic that is relevant for Africa. We don't want to guide you on which topic you want to do. This is where I also call Lydia saying, okay, I take you as a postdoc, just tell me what you want to do. I give you money and you can fly. So this was what we call the African Institution Initiative. I will not come back on the reason why research support or capacity building is very important. It was to handle the transition period for young PhDs and to make sure that we build the environment for young researchers who have finished their research to come back. And by that time, the most difficult part of our institution was that we did not have a postdoctoral position in our universities. This was the first time we tried to introduce, and we faced a big challenge at the universities asking us, where do you want us to put those fellows? They are not recruited as assistants, and they are just hanging like bats, if I can say. They are not attached to that. And some of them were saying, no, we want this position. We said, if you want this position, you need to reduce the time of lecture, because this program is for science. You need to take 80% or 90% of your time for science. So this was the big challenge, and we were happy that just after, I think Gordon knows the system to understand how best Africa can be supported. And this is where in 2016, together with the 11 consortium, we decided with the support of the funders to build the ASH, this think tank program that I'm coordinating. So ASH is made of 20 consortia coming from, I think, 16 to 17 countries, and we have about 100 researchers that are part of this network. But the 14 directors of the consortium form the ASH, and we are established to support all funders, to support all kind of research challenges in Africa. Because if you look at the group across Africa, we claim that we have the legitimacy to talk about what is health research in Africa. Just taking WACCBIP here, if you see the impact of WACCBIP and you multiply that by 14, you will see how the impact is in in Africa. We also help the funders to understand how, where to target investment and where to build the capacity. And the proof of concept of deltas is to shift the center of gravity of science or research into Africa, but not only research, but the funding mechanism and the management of research. This was the challenge that we had also for the trust from the funders to give this amount of money to African institutions without any good governance system. And then we have to look at areas of building the capacity. The capacity was not only for fellows, but it was also for institution and administration, and especially the financial officers. And you will see later, I will tell you, it's not only science capacity that we need, but we need to build the capacity of other part of the management program. And the model is based, as I said before, on the African defined priorities. What are the main priorities? Supporting institutions and people within the institution, and using also the tools. And if you look at this graph in blue, you see the program looking at different capacity building aspects. And we have some programs who help to develop tools, those programs who are deeply into the health research and health priorities. And this was something very important. And when you look at the 11 or the 14 consortia, you may ask yourself, is there competition among ourselves? But for us, it was not competition. It's to look where we can cross-fertilize, have synergies, and share platform. Sometimes when I'm here and I see fellows from WACCBIP, they think that I'm part of WACCBIP because of we were just exchanging. And when Gordon goes to Cote d'Ivoire or wherever, and the fellows see them with Lydia, they think that we are he's from Africa One. But for us, being part of one or another is not the important, but how we share resources, how we share skills. This is the most important part of the business. So what have we achieved so far? As I said, we moved from 11 to 14 consortia. And when we look at the figures, in 5 to 6 years, it's more than 2,000 fellows trained by the Deltas program in Africa, with more than 1,000 research networks and 5,500 undergraduates. And if you look at the network, you will see how our network is established. And you may see that the challenge is in Central Africa. And that was something Deltas was thinking, and for the current phase, we made possible to embark institutions from Central Africa into the program. It's 1,400 publications that we had. And if we take the funds leverage on top of what we did, each of the consortium mobilized 3 to 4 times the grant. I had 6 million euro for Africa One as Aspire. And we, at the end of the program, we were around 36 million, because this grant helped us to attract more grant by our fellows. And we have very strong institutions. And what is very important for this program is the balance between sectors and the balance between countries. Because sometimes when you build capacity, an ivory tower of an institution or center of excellence somewhere, and other countries or institutions are not following, it's a big problem. And you see what's happened. I'll just give you the example of Sierra Leone, Liberia, and Guinea with Ebola. Without capacity building in that area, when a disease emerges, you have it on your face. And to tell you, for example, today in Liberia, you have, I think, 1 or 2 vets in that country. Only 2 veterinarians, and they are occupying the high-ranked political position. So in the ground, you don't have people. So you see, if you take that into research, you can have very strong institutions doing research, but if you have weaker institutions or weaker countries, it's a problem. So Delta was trying to establish this network to help. And we have contributed during the COVID, I will not come back to that, how science can contribute to better prepare pandemics. And we had a paper on that. And we have also developed through the case of 4 consortia, how to really monitor and evaluate capacity building. We can monitor and evaluate research, but monitoring and evaluating capacity building was a big challenge, and we tried to put some tools and methods for that with the help of our partners in the North. And that gives us now a very good system of improving the evaluation and setting targets. The challenges level. I remember when Lydia wanted to go to a conference, I think that was the first time I realized that I have to pay for the baby, for the flight. I have to recruit a nanny during the conference for the child there, and I have to squeeze the program so that there are very specific time there that she can she can work. Just to give you an example. And sometimes funders also focus on the micromanagement, what I call micromanagement. Managing funding, good managing funding is good. But sometimes we forget to think that research evaluation is also very important. And the last thing I want to highlight is the mobility of researchers. Visas. It's a nightmare. And I don't know, maybe Gordon, we need to do something. We have been talking about that. This is where we need institutions like ECOWAS or African Union to help. I cannot understand that if I want to go to Kenya today, I have to take 2 weeks of application of visa, not knowing if I want to go or not. They cannot call me on emergency on something in a country where I have the skill and I can decide the following day to go. It's happened during COVID and Ebola, where when we wanted to bring East African social scientists to West Africa, it took a lot of time. So those kind of things are challenges, and we need to think of that. And one thing I want, as a mutual learning, is the area of administration overheads. And sometimes we struggle with funders. And the second element is keeping the funding foot in Africa, because we lack science funding mechanism in Africa, and we still depend on external funding. And what I want to highlight here is the freedom to look at or to address African priorities, and especially health problems that Africans face. And now I'm going to tell you the example of the One Health approach that I sold in 2008 when I was applying for that grant. So the One Health approach and how it can help the fellows here and even practitioners. So One Health approach, if you look at Africa today, we have more than 100 outbreaks ongoing. I don't know if in Ghana here you have the one dengue. Do you have dengue here? Ongoing dengue. So what are we doing on dengue? And you look at those kind of diseases that we can see, we listen, but there are others that we don't even look at. Food poisoning, antimicrobial resistance, malnutrition, mental health problem, those kind of health that every day affect people are not considered. And if you look at the health threats as a complex system, we have two ways. Either we use our tool, the tools we have in hand, which is not effective, and on top of that, you can also have the different perspectives, how we see the problem. The perspective of the communities, the perspective of natural science, and the perspective of humanities. So the most important things that One Health address is the collaboration between sectors, between disciplines, and then looking at the knowledge generated by this system to transform the knowledge with the contribution of the communities. So this is what we call the transdisciplinary approach. And why One Health? Because we see that if you look at the biology, you look at the physiology, you look at the pathology, human and animal share the same ecosystem. And there's no difference of paradigm between the physiology or biology of human or animal. They share the same pathology. And if we want to handle some of the diseases, we have to look at both sides to understand the issue. Then we ask ourselves 4 main questions. And the first one is how those threats emerge, and how, for example, the pandemics are fabric, and what is the cost of those pandemics or outbreak on the the livelihood of people. And the third question is, what is the capacity of our institution, our health system, to handle them? And the fourth question, do we have innovative solutions or approaches to address them? And if we look at the emerging diseases, some of the diseases from animal to human, we can definitely see that it's possible to handle them in the livestock sector to prevent human health. And I give you this example so that you see. This man is a physician. We were working in the northern of Mali. And if you want to look at the health of this child on the medical perspective, it's okay. And then another component will be, what will be the behavior of this woman to accept the type of medication you bring for the family? You can stop there. But with the One Health concept or approach, you can understand the health of this child by looking at the livestock, the source of their nutrition, which is milk. And some will stop with the nutrition, but those will go even far away looking at the grass available for livestock, which increase the milk production. So if you want to look at the health of this population, this pastoral population, and you only look at the child, you will miss a lot. So it's important to look at the global perspective of the environment of the patient, because everything is interlinked. Then comes to the failure to look at this holistic approach. You fail to look at this holistic approach, and if you look at the incidence cost with the time, this red curve, you will see that when an outbreak emerges, the cost is very high. We have estimation on that. If you look at Ebola, if you look at COVID-19, the cost of COVID-19 was very high, but it was possible in the wildlife sector, in the domestic animal sector, to make a surveillance and detect and respond quickly. If that was done, we could have reduced the cost. And the objective of the One Health is to flatten this red economic costs by using different mechanisms. We have the International Health Regulation that sets 19 priorities, the performance of veterinary services, and then looking at the condition with our contact with wildlife and the wash area to prevent the disease before it becomes a big problem. And I give you an example. Since we are here in WACCBIP, you work on AMR, antimicrobial resistance. And if you take a chicken, I think we had at lunch chicken. And if you take one chicken sold in the restaurant, the farmer gets from the price you have on the chicken in the market, the farmer gets 13% of the share. 13% of the share. But who do we ask to make effort to reduce antimicrobial use? Is the farmer. So if you put a social scientist there, what will be the willingness or the motivation of this farmer to prevent the health of people at the expense of his or her business? This is where the challenge is. The same thing happened with milk. Look at what farmer get from 1 liter of milk per day. This is the case in Mali. And if that farmer use antibiotics in 1 or 2 cows in the farm, and you ask the person to not use or sell this milk, if you have 5 liter or 10 liter per day, you see the amount of money the person will lose. So bringing this perspective to the problem will help to see how best we can improve the condition, the livelihood of people in order to protect the human health. And One Health is really relevant to Africa due to lack of resources, and we have demonstrated through different studies that you gain 14 out of 1 investment in using One Health. And one of the main problem now, if we take rabies, we know that rabies vaccine is effective, very good vaccine. It can protect, we can stop the transmission of rabies by vaccinating up to 70% of dogs. But now the problem is not in science, it's in decision. So we can move from the social science up to the political level on decision, how to make decision on, and I can give you more example on the added value of the One Health. So this is also one illustration of One Health. I borrowed this from Jürg Usinger from Swiss TPH. You see how our health system function with mass drug administration every year. It's good for business to sell the drugs, but all the time, every year, people get reinfested. So what do? Do you only rely on mass drug administration, or you need to add sanitation, good quality water? So those kind of things we are improving. And with that, we have trained many fellows in One Health, and we have a massive open online course on One Health you can attend. Every year we organize that in October, and you can attend this course with coaching in October, but out of October you can still attend, and but without coaching. And we can get a certificate at the end of the training. My last message is this picture. The first one representing, I will say, maybe fellows who want to capture everything. We are not really focused. We need to focus. And it also represents researchers in Africa. If you are a researcher, it's not like in Europe you have your lab and you know what to do. In Africa, you have to handle administration, you have to handle social life of people, you have to handle many, many other things. This is a researcher. And at the end, they will evaluate what is your impact in science. And the second message is supporting women in science. This capacity building is also there. When the women are showing their steps to grow, we need to look at social barriers that are really bringing them and breaking the glass ceiling. And the last one, I don't know if you realize, the seniors sometimes are not very good in helping younger ones to grow. This is, this, Gordon, am I right? Sorry. I missed the acknowledgement. This one is very important. What happened? The last slide. Okay, I will leave you with the conclusion, is this one. Those are the champions. I think Gordon is our leader in this. I'm coordinating, but we know that when we look how things goes, is the real leader, and I thank you very much for inviting us to here.
Thank you very much. Thank you very much for that wonderful keynote talk. In fact, when he was talking, two things came to my mind. The first one, for those who are biochemists here, is a signal transduction cascade, right? Phosphorylation and dephosphorylation. One small grant opportunity from deltas, or should I say one big, has evolved to something massive. And now we have this Arch platform, which is fantastic. And the second thing is really about the crosstalk between disciplines when we are conducting the kind of research that we do. We tend to focus too narrowly on only the area that we are more important, should I say, intellectual in. But I think we need to start talking across disciplines and formulating our research questions more broadly, so that when we are done, we'll be able to communicate the science that we have done to the right people who need to hear it, whether it's policymakers or even the communities or even educators. So with that, I think I would like to open the floor for questions. We have some time for questions. Yes, at the back.
Yeah, sorry. Thank you very much. So I believe you spoke on capacity building, but are you also looking beyond capacity building, transferability in science research? So beyond just doing the research and bringing the research to life, are you also looking at that? You also spoke on a bit on communication, science communication. There are times that I've interacted with people and then they complain that, scientists, the way we explain things to the communities, the lay people, it's a bit technical. So what advice would you give to young upcoming scientists in that regard?
Thank you very much. Very good question. I went very quickly. This is part of what we call the soft skills. But beyond the soft skill, we have one scientific foundation of One Health is the transdisciplinarity. What we do, and sometimes we have problem with funders on that aspect. When you recruit a fellow in Africa One, the first thing we do, I think we have Africa One fellows here, some of them are here. The first thing we do is tell you, go to the target population, not only the communities, but those who are going to take your research, like the national program. If you are working on TB, you need to go to the TB program and talk to them, talk to the communities. This is the starting point. Don't wait until you finish to start engaging them. And there's in Delta's program, what they call capacity community engagement grant that they put on top of the Delta's initiative. But in our program, Africa One, we already have that, which sometimes is not seen as purely science, but which help for policy and communities to take what you bring. If you wait until the end and you come with your result, the probability that is taken is very, very low. And we know the adoption rate of any innovation, the adoption rate is very poor. So if you want to increase the adoption rate, is at the beginning you have to set the research question with the people. And the second aspect of what we call transdisciplinarity is when you work with other disciplines and you generate knowledge. You generate knowledge from biologists, from chemists, from whoever, and you have a scientific knowledge. The second step is to put that knowledge into the community so that you take also the communities or the decision.
And also that WACCBIP epitomizes. In your experience, is this culture widespread at the moment? That's my first question. And secondly is that for Africans in diaspora who are still thinking of some way of engaging with back home, are there other avenues aside the Delta program and others? Are there other avenues where that's available at the moment for those who are still contemplating coming back? Thank you.
Yeah, it just remind me one researcher in Switzerland saying, I want to work in an area where there's no malaria. And I said, in Ivory Coast, there's nowhere you can have, yeah. So we have problem. The context of Africa is problem, we know. We also have potential. But here what we advise, nobody will, at the beginning, country, but I came back and I started setting my one health lab, and you see. Ask today. I don't know which country to take. I just take my country that I love, like Ghana, but I'm in Ghana, I will not say that. If you ask the Malian government where to put money, and you put research and to buy arms, what are they going to choose? I think it's the priorities, but it's our responsibility to show that science can help to handle some of the problems. And we need to show them also, or to discuss with them on different mechanisms of funding. For example, in Cote d'Ivoire, we have tried, I was explaining to Kevin Marsh yesterday, we tried to set up a national science foundation. What do? We had the debt given back from Switzerland to Cote d'Ivoire. And we said, okay, we want one share for science. And they said, no, we cannot prove that this money will really yield development. And we said, okay, we want this money, but it's your own money, and we have we put it as an endowment fund. And the interest that is generated every year is used to enroll 16 to 20 fellows, masters and PhD fellows in the country. And after 10 years, when we have evaluated, the country realized that the number of fellows we are training is gaining in momentum. And when we had the evaluation, the Ivorian government decided to put another $12 million into that. So doubling the portfolio of this grant. And then we have now an established science foundation where we ask funders to put money into that pot, but not diversifying it. And we call it what the wedding gift scenario. Do you know that? If you are having a wedding and you ask people to bring you gift, you may end with maybe 10 coffee machines. But if you have a list in the shop, when somebody pick a coffee machine, so you'll have to choose something else. So we have to bring funders to choose areas where we still have problem. Money is there, but money is going in the same place.
I think we have time for, just one more question. And the mic is already, okay, well, maybe two. Yes.
All right, good afternoon. Okay, so I'll start with our sister's question about science communication. By way of that, we have a show called Science Digest Show, of which Prof. Gordon Awandare champions for us as well. So we want to say thank you to him. We talk about science and health. We try to demystify science and health over there to the populace. So you can subscribe on YouTube, Science Digest Show. I have a question about collaboration, Prof. Yesterday, Prof. Ernest Aite spoke about collaboration. You also talking about collaboration efforts here. I want to know, how challenging has it been? Could you share some practical experiences about how to go about that so that we can leverage on the strength of each other to push the scientific community forward? Thank you.
I think collaboration is a culture, and we have not been taught how to collaborate. Our academic system is built in a way that we don't talk to each other. Look at how faculties are set up, and sometimes we don't have inter-faculty collaboration. And this is what we are building now in the academia. And my first PhD student who did the work on One Health with collaboration between different sectors, when he wanted to defend the thesis, it was almost rejected because they said it was not science. They don't know from which discipline he was. Those are the kind of thing we have as a problem. But it's this kind of research that can solve the society's problem. Not only a single discipline knowledge. A single discipline knowledge cannot solve a problem unless you mix that and then look at the area. And for the communication, I agree with you. We need to learn also how to communicate. But communication starts not on only one way. As I said, it's a transdisciplinary process. It's an iterative process. And I have seen that now there is a big interest of companies, small companies in science communication, which that help to take science to the society. And it's a very big component in our system now. And if you look at how others communicated on COVID-19, they were even stronger than scientists. So we need to make efforts in that.
Whilst we are on the topic of science communication, if you all have your phones, please go to the WACCBIP handle. There's actually a graduate student of WACCBIP who is a science illustrator at the same time. And for every single talk that you've heard, she has illustrated this very, very beautifully. Take a look at it and please retweet. This is ScienceCom live as it goes on. Good. We have one more question and then I'll end the session.
Thank you, Prof, for your beautiful presentation. My question is related to the last picture. The senior who is cutting the scale to go up, how will he get back? Don't answer that. But what can we do?
How? Repeat. Repeat.
Should I repeat?
Yes.
I said the last picture.
Yes.
We had a senior going
who's
up
cutting the steps.
And cutting the steps. I was asking how will he get back when he got all the way to the top. But my reflection point, please.
You know. Thank you.
Prof. Bofo, please.
He will fall. He will fall.
That's all. That's
how,
a very simple.
how can we change the mentoring culture in Africa? It is very important. You're giving funds to us to do research, but we are confronted to very poor mentoring. We have to humanize the mentoring in Africa, and I think each consortium has to take this into account. Thank you.
Yeah, I think it's a good and valid point. We take that into account, and it's part of monitoring and evaluation of capacity building. So being a good scientist, I think yesterday it has came, being a good scientist is not only the number of papers. We want to know how many fellows you have trained. Not only train, what are they doing? And when you move, who are at your back? Or when you retire, who will take over? And sometimes we have some of seniors retiring without anybody taking over. So those are the kind of thing we should put in the evaluation system of research.
Okay, so I'd like us to please acknowledge and thank Professor Bonko. Thank you so much. And next, we are going to move on to the plenary 7, and the session 3. The title of that session is Drug Discovery Mechanisms and Resistance. I'd like to call the session chairs Dr. Evelyn Yaria-Boni. Is it from? Did I miss it? And then Sebastian Arthur. Thank you. Thank you very much.
So I want to, even as we have session chairs, I just want to steal their job a little bit to introduce the next plenary speaker. I've known Thibaut since 2011. We're in the same lab. And now we are collaborating on a grant that he's leading. So I thought it was very apt that he would come and share some of the work that he's been doing with us. So sorry, Evelyn and Sebastian, but I'd like to call Thibaut to present his talk.
Thank you. That's a tough act to follow. We are going to go from the broader perspective to the very narrow perspective of HIV drug resistance, but I hope you will find it interesting. Thank you. So I'm going to have a fairly technical talk about HIV drug resistance. If you feel that you fall asleep or something, you can stand and walk around. And I will focus on a specific drug which is called dolutegravir. So dolutegravir is an integrase strand transfer inhibitor, and it's now part of the WHO antiretroviral drug regimen recommended for everybody. And one of the reasons for this broad recommendation is that essentially it's very, very difficult for HIV to become resistant against dolutegravir. So we say it has a very high barrier to resistance. And we did actually fairly a good amount of work with Peter, including on this topic particularly. And so that's one of the stones that we put in this wall. We studied indeed the barrier to resistance of dolutegravir, and it's indeed very hard for HIV to evolve resistance against this drug. And one of the reasons is that the resistance mutations that are coming up against dolutegravir have poor fitness, and so they are poorly replicative, and they have low levels of resistance, which led us to talk about an evolutionary dead end for the virus, whereby HIV cannot evolve high fitness, high resistant viruses when people fail with dolutegravir. However, we also had concern that maybe we're missing out on something, because this is true for the results when we sequence integrase, which is a target gene of this integrase inhibitor. In the Domono clinical trial, so Domono was a clinical trial that was led at Erasmus University, and the idea was to test the current regular antiretroviral regimen and compare it to dolutegravir monotherapy. Because dolutegravir was performing so well that people thought maybe we can just use it as a single agent. It actually turns out that you cannot and you should not, but in the course of this trial, one of the patients who failed was actually developed a mutation in this, what is called the 3' PPT region. And you can see this change. Well, you cannot see it, but you have to trust me. It's published, so. And there is this G stretch in the 3' PPT that was changed. So, of course, I'm sure a lot of you don't know what the 3' PPT is, and that's where it's a bit technical. So 3' PPT plays a role in the reverse transcription of HIV. The reverse transcription of HIV looks complicated, but it's very simple. You have the initiation here with a tRNA primer, and then you have the production of this strand of DNA from the RNA template, and then you have what's called a strand jump, and then you have production of another piece of DNA that is illustrated here. And during reverse transcription, another thing happened, which is a degradation of the RNA template. This is done by RNase H. And the 3' PPT is actually this part here in blue. It is resistant to the degradation by RNase H, and therefore it's a piece of RNA that remains after the synthesis of the other strain. And from this RNA can start to act as a primer for the production of the other DNA strand, which is this one. And then there is another jump, and you get eventually a double-stranded DNA that gets integrated in the human genome, and there you have a real infection. So the way we understand the 3' PPT mutations is that because this region is mutated, it cannot serve, it's not resistant against RNase H anymore, cannot serve as a template for the initiation of the second strand of DNA, and therefore we have the creation of circular forms of DNA called 1-LTR circles, which are therefore immune to integration inhibition because they simply don't integrate, but they readily can lead to the production of new viral particles. So the integration process is bypassed by these mutations, and therefore dolutegravir should not be active anymore. The only question, of course, is that this is true for some of the 3' PPT mutations that have been discovered or explored in vitro mostly, but not for the mutation that was found in the Domono clinical trial. So at this point, we still don't know, are these 3' PPT mutations a real problem, and do we need to monitor these mutations in people who are failing dolutegravir, as this medication is rolled out now on the continent, and that is used for nearly 16 million people. So we wanted to address this question, and we did this in collaboration with Brazil, and we were initially very interested in non-B subtypes because, of course, they're dominant in the non-Western world. And so we approached Brazil with a particular interest for non-B subtypes, and we tried to collect samples from people failing with dolutegravir, and then we sequence their 3' PPT regions, and we look for resistance mutations. The way the trial, this clinical trial was designed was actually flawed a little bit because there was no baseline sample collecting. So we still were able to identify series of 3' PPT mutations, and then to characterize them, we use methods that you saw a bit yesterday, whereby we can introduce them in models that we can transfect, and we can produce viruses that contain these specific 3' PPT mutations, and then we can test for their infectivity and resistance. So this is the only result slide that I have, so it can be relieved. These are the 3' PPT mutations that we found. We found these mutations in about 10% of people failing with dolutegravir. However, when we did the characterization, you can see here, this is a measure of the infectivity. So the higher it is, the lower the infectivity is. And this is in maximal infectivity compared to the wild type. And then you have the fold-change resistance against dolutegravir, which is here. And in green, what we found was actually often hypersusceptibility to dolutegravir inhibition. And there was only one virus that had minor resistance change against dolutegravir. And a lot of them were actually slightly impaired in viral fitness. So what does all of this mean? It's very simple, is that apparently these 3' it would be still very important to study their prevalence at baseline in people who were not exposed to dolutegravir. And there are still some. And with this, I'll be happy to take some questions. Thank you.
Thank you very much for that insightful presentation. Please, we have time for 3 questions maximum. So.
Thank you, Thibault. Actually, when I was reading your abstract, I was quite surprised because I think a few years ago in Bordeaux, there was this investigational drug that when they do the drug selection, the resistance selection, they were selecting mutations in the 3PT region. And they saw that that was conferring resistance in the virus. So, I mean, maybe I haven't, I'm just wondering, how does this fit in line with that? I think it was Anne Marcelin's group.
Yeah, so that's a good question. So that not all 3' PPT mutations are equal, essentially. So if you have deletions in the 3' PPT, including outside of this G stretch, and specific mutations in the 3' PPT in the G stretch, then most likely what happened is that you indeed go towards this pathway of 1-LTR formation, and therefore you can develop resistance. But apparently this is probably not a process that happens very readily in people, and that's why the mutations that you find in people versus what you can do in vitro or in cell culture, essentially the mutations are different. And I would be surprised to see the mutations we see in vitro in people, to be honest. Yeah.
Okay, thanks.
Any more, please? I can't seem to see any other. Okay, right here, please.
All right, thank you very much. I want to believe the mutations in the PPT region leads to
or avoids or prevents the integration step from happening. How does this affect reservoir?
And produced in a different process. But these 2 LTR circles can be found in people under treatment for more than 10 years. So before we used to think that they are a byproduct of abortive infection and they are essentially garbage. But they are not. They are probably potentially another source of rebound. This has to be tested. But definitely the HIV reservoir seems to be more complex than just integrated DNA. Whether these 1 LTR circle forms are also part of the reservoir remains to be studied, because there is no way, or it's quite difficult to distinguish them from 2 LTR circles or from integrated DNA. So the molecular tools are not there quite yet to study this, but we're working on it. So yeah, we will try to address the question. Thank you. And thank you again for the kind invitation. And thank you, Peter. I see you're not listening to me anymore. No, I'm joking. So, but I'm also very happy to be here. Thank you.
Thank you very much.
Okay, that was indeed technical. We'll go to the follow session. We have our first speaker, Mark Apienin, who is currently characterizing the HIV cohort in Ghana. He's going to talk about his contribution towards HIV cure research.
Thank you very much. My name is Mark Apienin, and I'll be talking about characterizing an HIV cohort in Ghana and its contribution towards HIV cure research in West Africa. Having listened to a very broad talk and a very narrow talk, I hope mine will fit into the middle so that we can all flow with me. Thank you very much then. So HIV has been with us for the past 4 decades, but still continues to be a disease of public health concern, with about 39 million people living with the disease as of 2022. Number of new infections continued to decrease, but in 2022 we had 1.3 million new infections, with a little over 630 million. Dolutegravir has a high genetic barrier, which we've heard before and, in our previous talk, and the fact that it doesn't require any boosting. And also, it is known to have limited cross-resistance to other first-generation integrated strand transfer inhibitor. Let me say that dolutegravir is the first of the second-generation integrated strand transfer inhibitor that was approved for use by the US FDA in 2013. Again, in Ghana, since with the introduction of the dolutegravir-based regimen, we do not have a lot of data on how well it is doing within our community. So it is instructive to know that the virus that causes the global pandemic, that is HIV group M, has a lot of subtypes, and these subtypes are distributed, are found within different geographical areas. In subtype C is the predominant subtype found within Southern Africa, whilst within Western Africa, what we call the circulating recombinant form O2 is the predominant subtype of HIV found within West Africa. In Europe and most of the Americas, we have what we call the subtype B, being the predominant HIV subtype within these geographical regions. And most of what we know about HIV has been on subtype B, because that is what is found in most of the most European countries and in the Americas. It is instructive to also know that there are differences in the genomes of these various subtypes, and these may have impact on the response to antiretroviral therapy. Indeed, it has been shown that some mutations, or these mutations have different impacts in different subtypes, or the integrates of different subtypes in culture. What then do we do? Having known that we have different subtypes in different places and may impact the response to HIV, it is instructive, or it is essential that as we look for new treatment, as we look for new cure for HIV, it should be geographically inclusive. And that is why we are embarking on this study. The rationale is that we believe that a well-characterized cohort in Ghana will be essential, as it will help us determine the impact of various drug regimen in Ghana. It will also provide us the opportunity to undertake cure research, cure-related research, including clinical trials. Then, more importantly, like I've said, most of what we know about HIV has been on subtype B. But what we have, the predominant subtype in Ghana, is the circulating recombinant O2. And this cohort will provide us the avenue to understand exactly the local epidemic. Some cohorts elsewhere have given us the opportunity to understand HIV. For example, this Capresa cohort in South Africa has given us a lot of understanding to HIV in South Africa. That is the subtype C, and a lot of clinical trials have been embarked on in South Africa because of some of these cohorts. Additionally, this also fresh cohort in South Africa has also given us the opportunity to understand the immunology of hyperacute infection in South Africa. So it is important we have something like that. So with this work, we aim to establish a long-term HIV infection cohort in Ghana. And to do this, we recruited people living with HIV and comprehensively characterized them. We also studied the cellular immune response within this cohort. Then again, we evaluated the impact of dolutegravir-based antiretroviral therapy on the viral load within this cohort. This is how we went about it. We obtained ethical approval from the Ghana Health Service and ECBAS. And within this cohort, we have two categories. We had the naive, ART treatment naive group, which we followed them up every 6 months. Then we had another group, which were the ART experienced, which we cross-sectionally sampled them. Then we have so far sampled from four hospitals, 1 in the Volta region and 3 in the Greater Accra region. We are looking to hit a target of 1,000, but we are not there yet. So we continue to expand. So the first part was to establish the cohort, and this is how we went about it. We recruited the participants being both ART treatment naive and ART experienced. Then we reviewed their medical record, administered questionnaires, we drew blood sample, we continued to do HIV typing, we did some screening for sexually transmitted infection, including hepatitis B, C, and syphilis. We did hemoglobin estimation, then we looked at their liver function, we looked at their renal function as well. So far, we have been able to recruit 380 participants. We have 153 being ART naive and 227 being ART experienced. And these are the contributions from the various sites, the highest coming from the University Hospital here in Legon. By way of age distribution, we realized the majority of the participants are within the 15 to 49 year age limit. The implication is that these people would have to be on treatment for the rest of their life until we find a cure for HIV. And these are the working, majority are the working group. These are the modal responses for the various characteristics that we try to look at in trying to characterize them comprehensively. We have gender, we have the educational status, we have the occupation, we have the marital status, and we have the possible mode of transmission. It is instructive to know that within this cohort, the common transmission route is through the heterosexual, and this is most common in all other West African countries, as compared to the West, where it is through mostly homosexual. We looked at self-reported conditions, and we have 13.7% reporting of reporting having a chronic condition, hypertension being the most common. Then we looked at the ART regimen on which these participants were on. We realized majority, 97.6%, are all on ART, the dolutegravir-based regimen. And out of those who were already on treatment, some had a change in regimen, and mostly it was because of the change at the national level. That was in 2019 when we adopted the dolutegravir-based. It's 0.5%. Within our cohort, I must say we have 13 pure HIV-2 participants and 44 who have dual infection. Anemia was prevalent within this cohort. So then in terms of, we looked at the trend, the response to, we looked at the response as, we looked at their responses by measuring their hemoglobin, and we realized there was increase in hemoglobin concentration from time as they started treatment. And we looked at whether there was a damage to their kidneys, but we realized that at the beginning we didn't see any damage. But with long-term treatment, we see there's a reduction in the eGFR. Then we looked at the liver as well. We realized there was no significant, there was no damage to the liver as we see. Okay, there was no association between treatment status and the and renal function, as well as the liver. So we say that we have been able to establish the cohorts, and we continue to expand on the majority willing to participate in any future clinical trials. The second part was to look at the cellular immune response, and to do that, we looked at the CD4, CD8 T cells, and we estimated them from whole blood using a two-color flow cytometry. We say that there was a significant improvement in the CD4 count after 6 months of treatment, and even to the recovery to normal levels within those who were ART experienced. For the CD8 T cells, we realized there wasn't significant differences for the treatment naive that we followed up, but between the decreasing with treatment, but it wasn't decreasing at the rate at which we expect to see. Okay, so we had a lot of participants still having viral non-suppression within this cohort. So in summary, we are saying that there's improvement in dolutegravir based. There's improvement in the viral load, but then it's approved or some verified kit for typing. So in our case, we use the SD BioLine for the subtyping, for the typing. Yes.
All right. Okay. Thank you.
I'll spare that. Yeah. Please go ahead.
Okay, so there are a number of things that can be we can look at. It could be we are yet to find out if there are drug-resistant mutations within the group. That could be one. Whether there are drug-resistant mutations in there, or there could be the fact that people are not adhering to the treatment, which is another arm that someone else is looking at.
Okay, I have 2 questions, okay, but I'll ask you later because your time is up. Thank you so much, Mark.
Thank you, Mark. We have our next presenter, Abigail Obobi, representing Unveiling the Plasmodium Inositol Phosphate Pathway, highlighting IPMK as a novel therapeutic target for malaria. Abigail, you have 15 minutes.
Thank you very much for having me here, and welcome to malaria. Okay, so we are back to malaria. So this work is part of my PhD work, and the foundation of my work is we know the problem with malaria now, that there's a development of resistance towards very effective antimalarials. So my vision was to find novel ways to target malaria. So I'll start with the pathway. There's an interesting pathway in eukaryotes called the inositol pyrophosphate pathway. And this pathway involves the sequential phosphorylation of inositol trace phosphates, popularly known as I3P2, inositol hexacase phosphate, and inositol octacase phosphate, known as pyrophosphate. The players in this pathway are kinases, and we all know kinases are well-known drug targets. And for the purpose of my presentation, I'll be more interested in the inositol polyphosphate multikinase (IPMK), inositol pentacase phosphate 2 kinase, inositol hexacase phosphate, and diphosphoinositol pentacase phosphate kinase. My work too is a bit technical. I'll try to calm down as much as I can. So this pathway is an integral part of calcium and phosphate or energy homeostasis. And if you know the biology of falciparum, the most virulent causative agents for malaria, we know calcium metabolism or calcium signaling is very important. especially in synchronization, which helps the parasite evade the host immune response. So having known this, I did a little background, and I saw that the pathway is essential in humans. This pathway is essential in other parasites like Trypanosoma brucei, the cousin of Plasmodium, which is the T. gondii. I've seen the pathway essential in plants, and one popular metabolite of the pathway is phytic acid. We know even as humans, some people take that thing as a supplement. So having seen all this, I didn't see this pathway being studied in Plasmodium. And this led to the hypothesis where we hypothesized that the Plasmodium inositol pyrophosphate pathway is present and essential to the the erythrocytic Plasmodium parasites. And to answer this, we had 3 objectives. Basically, we dwelled on the powerful tool of bioinformatics and PCR to be able to characterize the genes, putative genes in this pathway. We confirmed the presence and also in silico chemically validated them. So the summary of the methodology I used, we sought ethical clearance from the Noguchi IRB. Then went on to sequence and structural analysis, PCR with sequencing. Then we used molecular docking to do virtual screening to find selective inhibitors to the kinases. So we were able to identify 4 genes which encode the kinases in this pathway in Plasmodium falciparum. The first one we call IPMK1, then we have IPMK2, IP6K. Then the last one is actually a multi-domain gene which encodes an enzyme called VIP1. So with the architecture of the kinase domain, with the architecture of the kinase domain, we could identify several intersections in the Plasmodium enzymes as compared to that of the humans. And this kind of confers special activity or special functions to the Plasmodium. It's popularly known actually in parasites. But with the sequence identity, we did a pairwise sequence identity, and we could see here that these enzymes actually, let me chip this in, they are broadly divided into 2 subgroups based on the functional motif. So we have the PDKG enzymes, and 3 of the 4 genes I studied were PDKGs. So I grouped them together here. These 3 are PDKGs. So you could see that they had very low sequence identity to the human enzyme. And the 4th one, which is the VIP1, had 44.7, which is a bit high. Then we proceeded to characterize them in terms of evolutionary relationship. So what I could simply show is that the genes, the protein, all what I'm dealing on now is actually the kinase domain, because I know when we talk about kinase domain, those are a bit specific than looking at the whole protein. So every analysis I did here, I used the domain, kinase domain. So if you look at the domain labeled in blue represents the Plasmodium PDKGs, and the pink one represents the human PDKGs. And we could see that the Plasmodium enzymes are analogous to that of the humans. And for the VIP, we could also see the Plasmodium, which is blue, that is the most diverged in the analysis we did. Then we went on to determine how structurally distant they also are. We know protein fold energetically, because of how energetically favorable they could be. So when checking the structural divergence, we modeled the structures of the 4 domains. Then comparing to the human, we know kinases have different parts. We have the C lobes, the hinge, the IP helices, where the substrate binds, and we have the N lobe. They are quite similar to the protein kinases. So when we did the okay, it's not moving. So we did the structural alignment, and the cutoff we used was the RMSD values, which it's not responding. The RMSD, yeah. So we did the structural alignment, and the cutoff we used was RMSD value of 2. So any alignment we got, the value we got, if it's below 2, it denotes homology. So we could see that both IPMKs here are structurally dissimilar and not homologous to that of the human, as compared to the I6PK and the VIP. So the other important thing we did was to because they are kinases, we know kinases bind ATP. So we wanted to now we have the same, two genes, which supposedly does the same thing. So we wanted to make sure that they are all okay. And as I said earlier on, in the identifying or in analyzing the sequences, we saw a mutation in the protein sequence of the IPMK1. So here I wanted to find out if structurally that mutation is going to affect the binding of ATP. So what we did was to dock ATP in. And we could see that we were expecting ATP to bind to the hinge region, which we predicted the residues to be valine and the glutamine. But the ATP took the binding, the mutation was able to take part in ATP binding, which is not supposed to. Meaning if this protein is even expressed or produced, it's not going to be functional. So we then moved on to, we moved on to the IPMK2, and we could see clearly that the adenine moiety of the ATP was able to form hydrogen bonds to its right predicted residues, which shows that the IPMK2 is not redundant. And it's essential because we've gathered data from the whole genome-wide mutagenesis studies, which shows that this gene is essential. So I now bring you to the pathway So this pathway starts from the cleavage of PIP2 by PLC, phospholipase C, to diacylglycerol and I3P. And we know from our classical biochemistry that I3P is involved in calcium mobilization from the intracellular stores. But IPMK is able to use I3P as a metabolite, which sequentially phosphorylates to I5P or IP5. And we have I6PK, which also is able to phosphorylate it further to a pyrophosphate. But what we saw in this plasmodium pathway is that it lacks the inositol pentakisphosphate kinase, which is supposed to phosphorylate I5P to I6P. So for this, we hypothesized that probably the parasite scavenges the IP6 from the host, or I6P is able to synthesize the substrate. And we know also from this, the I6P is also behaves as an ATP synthase, where at low ATP is able to synthesize ATP from ADP. When ATP is high, it's able to, the pathway favors the pyrophosphate production. And this pyrophosphate also is implicating several signaling processes, as well as acting as a metabolic switch. So as in if ATP is low, the VIP gene has a histidine acid phosphatase, where it can cleave off one moiety of phosphate to release energy to supply the parasite. And in the nucleus, we could see it was based on data in literature. The pathway in the nucleus is involved in signal transduction, epigenetic regulation, and all that. So one of the most important things we did was, because we were dealing with putative genes, wanted to find out if it's actually present in the parasite. So the F1 is the field isolate, 3D7 and DD2. We're able to find the parasites, the genes in there. So we went to the structure-based discovery. So we did molecular docking to find out if we could see inhibitors that have preferentially high affinity for the plasmodium enzyme than that of the human, and could identify a few of them. Then here is the fragment-based discovery. So we started with a library with very small molecular weight ligands, and we were able to find a scaffold. So the initial binding here, we could see that it had good interaction with the enzyme. Then we did a substructure search in zinc and other databases, PubChem and all that. They were able to find some agents which had a bigger molecular weight, which also showed preferable activity for plasmodium protein than the human. So here, I know it's a bit technical, but what I want you to take home is that we've seen an interesting protein that is not reductant, it has a sufficiently unique structure, it's not autologous to the human enzyme, and it's druggable. And the advantage of pursuing this is that it has a possibility for selective inhibition. It has potential activity across multiple life cycles, and possibly for like a possibility for pan-species activity. So we have seen, we have evidence, we have shown the evidence of the presence of genes encoding the inositol pyrophosphate pathway in Plasmodium falciparum. And overall, our data shows that it shifts the signaling from I3P towards the synthesis of higher inositol, which are energy rich, and we believe that is important in calcium homeostasis and phosphate homeostasis. And that if we target this pathway in Plasmodium falciparum, it may provide different or a new avenue for anti-malarial agents so that we overcome the evolution of drug resistance. Thank you. So here I acknowledge my supervisors, my Prof. Ben Kwashi, Prof. Nancy Kwashi, and Professor Sayers from the University of Sheffield, and all the lab team from Kwashi's lab and the Sayers lab. I'm very grateful for the support given me. Thank you.
Thank you very much. I don't know whether you succeeded in delivering on your promise of breaking it down, but the audience are in a better position to do that for us. So any questions for clarification? Yes, one there.
Yes, so my question is still on that. Can you please summarize your research to the watch seller at night market?
Okay. Okay, so basically, to the watch seller. Before, as Africans, I don't know of any Africa or any Ghanaian that have not had malaria before. And we know the effect of malaria. I didn't actually believe malaria could kill until I had a very close relation having a complications in malaria. I realized that it's deadly because I didn't understand why malaria should kill anyways. But once I experienced it, I know that it's deadly. So for the development of resistance, where we take in drug and it's not able to work on our system or kill the parasite, it will lead to more death. So my mission here is to look for new ways, which I feel like will be very important to the parasite. So if you take that thing away from the parasite, they are going to die. So the new ways I'm looking at to study the biology of the parasite and find agents that would inhibit or target that aspect of the parasite, very important to the parasite. And hopefully we can develop a new drug towards malaria, or against malaria, sorry.
Okay. Thank you. There's a question in front, please.
Actually, I was wondering whether you were referring to I3P or IP3 or NIP6. And then when I realized on slide 14, you actually put I3P.
Yes.
Should it be IP3?
Yeah, the nomenclature of these agents are very confusing. In literature, some people say I3P.
It's inositol.
It's inositol trisphosphate. So I feel that's why they write I3P. I usually pronounce it IP3. But when during my literature search, I realized they more use I3P, I4P, which even the pronunciation is difficult.
The way you write it means is you have 4 or 3 molecules of the inositol to 1 phosphate.
Yeah.
That's what you have written.
Yes. So as I said, that's what is in literature. But I felt it had to do more with the nomenclature, how they pronounce it. Like inositol tetrachysphosphate, inositol trisphosphate.
Please, one last one at the back.
Thanks, nice talk. Whether it's tomato or tomato, something to think about is even though it doesn't have targets theoretically in, let's say, human systems and cells with this particular signaling pathway, something that's worth doing as part of your work is stimulating some human cells with it and looking at some cytokine, chemokine profiles by hitting it with something perhaps like hemazone to just see whether or not it changes those profiles. So some of these drugs, despite their specificity, alter the immune profile in the human cells, thereby changing some of the expected actions. And it's a straightforward experiment to do, and I'd encourage you to do it. Thank you.
Thank you, too. Well noted.
Thank you very much, Abigail. Let's appreciate her.
So we managed to survive technical sessions. Yeah, I had a lot of questions, but I don't even understand the questions myself. So I'll just keep it. Thank you so much, our speakers. Please give it up to them. So our next session is the table talks, and that will be led by Dr. Peter Kwashi.
Let's thank Dr. Bonnie and Dr. Arthur for a great session chairing. So those of us who follow us on social media would have seen that we do a number of events during the year. And one of the things that we did few weeks ago, maybe 1 or 2 months ago, was that we had this vaccine science forum, and then we had a 2-day open science day here at WACCBIP, where we invited 7 high schools to come, and basically we exposed them to WACCBIP, what we've done, our impact. They went around the labs, they did some little experiments here and there were some demonstrations, and then we gave them a challenge. If they have some ideas, innovative ideas, they should send us those ideas and then make a poster of it that we'll present here. So we have students from 2 schools who will come and do a turbo talk to present the idea that they had. I haven't seen the idea, so I actually can't tell you what it is. And they're from the West African. Have they entered here yet? I'm preempting them a little bit. Yeah, but there will be students from the West African Senior High School, WAS, as is popularly known in Accra, and also students from Frafraha Community-Based School. So they have their posters on the first floor. When you walk in, it's up against the wall so you can see it. And this is our way of trying to encourage these up-and-coming young scientists, we hope. So they just arrived here from school with their school bags. So unlike, they don't have a one slide, but they'll come up and talk about their poster and invite you to their poster. So I'll ask the students from West Africa Senior High School to Senior Secondary to come up. Was, come and talk about your poster. And this is in the same manner. Oh, come. So you introduce yourself and you tell us about your poster and why we should come to it. And this is in the same manner as it's a turbo talk, basically 2 minutes.
Good evening, everyone. Please, I'm in the person of Emanuela Amanyako Nsiedu, and I stand before you today not just as a student, but as a survivor. A survivor of malaria. Growing up in Bonata, so that concussion recovered me. So it was then I realized the potential power of nature to heal. So it's artemisinin-based combining therapies, ACTs. ACTs. Her story ignited me. Her story ignited a fire within me. proving that even a woman from a humble background can make a significant impact. So that was where I got my inspiration. So Ghana, my beloved country, has eradicated anti-malarial drugs in order to make a malaria-free future for us. Thank you.
All right. So let's give her another round of applause. I think nerves are something that all of us experience, but she did well. Notwithstanding. So after that, I'll call Frafraha Community-Based School to come and present their idea.
Good evening, everyone. Okay, so I'm with Bolabi, and then I'm here with my colleagues to talk about a virus which has affected over 80% of the worldwide population at a point in their lives. So it's a leading. This virus is a leading cause of cancer in our daily life. So I would like someone to guess the type of virus I would like to talk about. Yes. So I'm referring to the human papilloma virus, which is commonly known as the HPV. So I would like to talk about a short story which inspired us to go for the human papilloma virus. So my auntie in her mid-30s was diagnosed of the human papilloma. She was diagnosed of cervical cancer. But fortunately, she received treatment and she's cancer-free now. So her experience and the pain she went through made us want to talk about the HPV, its importance, yes, and the impact it has on our lives. Even though the HPV is very common, it is often misunderstood by a lot of people around us. So I would like to talk about I would like to give a detailed presentation of the HPV when you come over to our poster. Yeah, thank you.
Okay. So we are the. Let's give another round of applause. And then we're going to call people up to come and give their turbo talks, which is a one slide. It's not every poster that got a turbo talk. So these were posters that were highly scored, and so they'll come and give a one slide and talk about why we should go to their poster. While others are being lined up, I'll call up the first two, Gloria Pokua Menu and Ernest Afari. And you have 2 minutes each.
Good evening. Okay. My name is Gloria Pukua Menu, and I'm presenting a poster on the topic predicting SARS-CoV-2 antibody binding to selected respiratory viruses. This is to explore the potential for cross-protection. So respiratory viral pathogens are a public health concern, and they cause respiratory viral infections. And a recent report on the leading causes of death shows that respiratory viral infections are the second leading cause of death in Africa. And this is quite an interesting report, because the morbidity and the mortality of respiratory viral infections in Africa is quite high. And so what is the problem? The problem is that there are no licensed routine vaccines for many of these respiratory viruses. And currently, only 2 vaccines are licensed for routine use: SARS-CoV-2 and influenza virus. Just this July, 1 vaccine was licensed in the USA for respiratory syncytial virus, but even at that, it's only for the aged. And so we asked the question, could the impact of COVID-19 have any beneficial effects for us? Looking at the extensive vaccination that has gone on globally, could we harness the benefits for control of these other respiratory viruses, since it is very important to look at alternative control measures? And so in this study, we tried to use in silico analysis to bind the antibodies of SARS-CoV-2 to other selected respiratory viruses. So for my poster, what we did was that we showcased the results for respiratory syncytial virus, and the results are quite interesting. We first looked at aligning the antibodies for the 2 different viruses to see if there are similarities or identities between these antibodies, and then went further to dock SARS-CoV-2 antibody with antigens from respiratory syncytial virus. The results that we obtained is quite interesting, and I would love that you come to poster number 13 and for a detailed description of what was observed in our study. Thank you.
Hello everyone, greetings to you all. Okay, I'm Ernest Afare, presenting on Plasmodium falciparum antigen-specific antibody levels and avidity of antigens in Ghanaian individuals across varying malaria transmission intensity. Humoral immune response are key in clinical immunity against P. falciparum specific antibodies or infection. In high malaria endemic regions, affinity maturation of P. falciparum infection is hampered in both children and adults. This study has been done on the blood stage antigen, which is the MSP3. It is unknown if antibodies against the pre-erythrolytic stage and the sexual stage antigens are similarly hampered in high transmission settings. So the study estimated antibody responses and avidity of antigens from the 3 different stages of P. falciparum specific antigen. That's the liver state antigen, CSP, and the blood state antigen, MSP3, and the sexual state antigen, that's PFS 230. We use an indirect ELISA method. 316 plasma samples were used, of which 174 were from Accra and 142 were from Cape Coast. Accra is demarcated as a low malaria transmission region, where Cape Coast is demarcated as a high malaria transmission region. Come to poster number 5, and my finding will shock you. Come listen, come and be educated, come and be imparted. See you at poster number 5. Thank you.
All right, thank you, Ernest. So I'm waiting for the next few slides to be queued up. So Chris, give me a thumbs up when you're ready. In the meantime, let's get up and do some stretches. I know we'll be walking soon, but let's take advantage of this. We've been sitting for a while. And so, okay. All right. Okay. So we're good to go. So let's get ready. I'm going to call up the Turbo Talks, Becky Ramatete. So you can line up, and once the first one finishes, the next one goes. Becky Ramatete, Precious Kwesi Kumi, Timothy Letza, Yinka Submarine. And then we'll take these for now. So I didn't realize they reordered it. Okay, Ernest, we've already. So Precious Kwesi Kumi. Is Precious here? That's not your slide. Theo, that's not your slide. No. Precious, you stay. So Precious Kwesi Kumi, Timothy Letza. Let me see. 1, 2. Lucinda Frainate, Eugene Lamte. Can we have slide number 1? Huh? This slide number 2. Come and talk to me. Come in. Okay, so let's do it like that. If you see your slide, come up and talk about it. I mean,
Hi everyone. Okay, so just imagine you're a bacteria, and then you have coat, and then you have different colors of this coat, okay? And whenever you get to wear one of these coats, you get to have a different identity, and that identity gives you a different level of causing harm without being caught. If I were you, I will always put on that coat, right? So I'm working with Streptococcus pneumoniae, which is a gram-positive bacteria, and I'm looking at how 8 coat, which is a polysaccharide capsule, together with 8 antimicrobial resistance, is helping it to evade its host without being caught. My poster is poster 4, and my name is Jennifer Morgan. So please come and hear more about that. Thank you.
Hi, I'm Precious Kumi, and I'm here to talk about COVID-19 booster immunity. Basically, how long did your booster immunity work? That is with respect to the COVID-19 vaccination that you received. Now, booster vaccinations will typically stimulate your immune system to generate a heightened response against COVID-19. Now, the quality of that response is affected by a number of factors, such as your immune status, your previous vaccination history, and then to a large extent by maybe the type of booster or the vaccination regimen that you would receive. Now, I'll be looking to measure T cell and antibody responses against, I measured T cell antibody responses against COVID-19, and then the goal was to determine how long your immunity lasted. So please meet me at poster 3, where we can have a lot more conversation about this. Thank you.
Good evening, everyone. I'll be presenting on cytokine profiles in asymptomatic malaria children with different hemoglobinopathies. Okay, so asymptomatic malaria remains a public health concern, particularly in regions where it is endemic and prevalent. This is because individuals who have this condition do not report the disease, and they contribute to the transmission of the disease since they don't report it. And also, genetic factors genetic factors like sickle cell disease are also associated with inflammatory responses, and these inflammatory responses are mediated by cytokines, which are proteins and soluble molecules that regulate the immune responses. So in my study, I measured or I assessed the levels of pro-inflammatory cytokines and also anti-inflammatory cytokines. IL-6, IL-8 for the pro, and also anti-inflammatory cytokines, IL-4 and IL-10, using the sandwich ELISA method. The samples were collected from 3 communities, Begro, Estuary, and Tomefa. So my study found out that individuals who were positive for malaria had significant levels of IL-10, indicating that indeed these individuals may be asymptomatic. And also, my study found out that individuals who are positive for sickle cell disease may be in crisis because they have significant low levels of the anti-inflammatory cytokines, which is the IL-10 and then the IL-4. Overall, my study contributes to the understanding of So you can come and meet me at poster number 15 so that we can delve deep into this. Thank you.
All right. Good evening, everyone. Yeah, so I'm trying to model malaria drug resistance, and yeah, so as we can see, malaria is a threat, as we already know, and it is expected that if this drug resistance that we are having currently facing, for example, the ACTs that are majorly used for drug resistance, if we don't look at this problem and how fast it will spread, then we shall have issues. For example, it's confirmed in East Africa, South America, and Southeast Asia. So what are we trying to look at? We are looking at linked populations of mosquitoes and humans, and we're looking at probabilities of events happening within the transmission dynamics of malaria. The major assumption we make is, what if it's easier for these drug-resistant parasites to pervade a population of people that are susceptible? What if it is easier for for the drug-resistant parasites to invade a population of people that are susceptible than those that are already carrying one mono infection? So we ask ourselves a question: does this assumption help us to understand the fact that resistance will spread faster in low transmission areas than high transmission areas? If you want to discuss more about this, I'll be waiting for you on poster 35. Thank you so much.
Hello everyone, my name is Lucinda and I'm presenting, my poster is on the impact of amino acid polymorphisms in Ghanaian HIV-1 post sequences on their structural characteristics, and this is an in silico study. So I'm sure we were all surprised and a little scared about the reports that was given in the news on the number of people infected with HIV in Ghana. So to jog our memories a bit, to give you that perspective, in 2022, Ghana recorded over 16,000 new cases. And these infected Ghanaians may resist antiretroviral therapies due to mutations in the HIV genome. So this study aimed to examine the impact of these mutations on proteins encoded by the HIV POL gene, which is integrates reverse transcriptase and protease. These are very key proteins that help the virus to survive and proliferate or thrive. Yeah. So we first retrieved the sequences from the Los Alamos HIV databases. So these are Ghanaian sequences. We used the Stanford HIV drug resistance database to perform a genotyping to identify the most prevalent mutations in these Ghanaian sequences. We then modeled these proteins and identified the effect of the mutations on their stability. We then did a molecular docking study with the known inhibitors of integrase, protease, and reverse transcriptase. And we then, so the molecular docking study was to see the change in the affinity for the proteins after these mutations have occurred. So we then move on to assess the interactions, the protein ligand interactions between the inhibitors and the mutated proteins. And then we do a molecular dynamic study to check the stability when these inhibitors bind. So meet me at poster 41 and let's talk. Thank you.
Good afternoon. My name is Eugene Lamte, and I'm presenting on a drug discovery project using machine learning. I'm presenting on a drug discovery project using machine learning targeting the herpes simplex virus, specifically the virion protein 16, which is and these machine learning models were evaluated on various machine learning various evaluation metrics such as the accuracy and other. Yeah. So different machine learning models had different evaluations because they have different ways of making classifications. And we were able to take the 3 best models and then deploy them using web servers. So at the stand, we would be able to give a live experience of how this, how the web server works and use the models to make predictions on new data. Thank you. Poster 55.
Hello everyone. So my name is Caleb and I'm presenting on determination of virulent gene diversity of Helicobacter pylori in Ghanaian patients with gastric dyspepsia. And this study was done here at WACCBIP. Okay, so H. pylori is a gram-negative bacteria that contributes mainly to gastric dyspepsia, right? And then in certain regions, genetic variability of virulence factors play major role in the disease progression or differences in clinical outcomes would be seen in Ghanaian patients with gastric dyspepsia. So what we set out to do was to investigate genetic variability in certain virulence factors of the Helicobacter pylori in Ghanaian patients, right? And then what we did was we in this study we took stool samples from the patients with the gastric dyspepsia, and then through targeted PCR amplification methods and then sequencing techniques, we amplified the Kage 3' variable region, and then we sequenced it, and then we did a multiple sequence alignment. So that's what we can see here. And then we had surprising results also. So please, if you want to know more about our findings, you can visit poster 73. Thank you very much.
Good evening, everyone. My name is Helen Kamasa. Then I'm presenting on this topic, bacterial vaginosis and correlates of cervical cancer in apparently healthy Togolese women. I'm happy that my work has been briefly introduced by these ladies here. So I want just to let you know that apparently healthy women can have anginal cervical cancer. And this cancer is the second most common cancer in women in Africa here, and it's caused by persistent HPV infection. But what is amazing is this cancer is the most preventable cancer, suggesting that there are other factors that are included or that are included in his risk factors. So you are kindly invited to visit me at the poster 77 for more interaction about my work. Thank you.
Hello everyone. My name is Becky and my work is on the functional characterization of H2 receptor in COVID-19 infected West Africans. COVID-19 pandemic is over, but it's still necessary for us to understand the dynamics that happened during the pandemic. And beyond the research that has already been done in malaria and looking at immune signatures, my work focuses on looking at host receptors that may have polymorphisms that may have led to the clinical phenotype for West Africans, which was predominantly mild. So we are hypothesizing that if people have mutations in ACE2, there's a probability that it may have a lower binding affinity to SARS-CoV-2, and that may lead to either severe, mild, or asymptomatic clinical presentation. And I have tested this using in vitro and in silico assays, and I would love to see you at poster number 83 to talk more. Thank you.
So, good evening, everyone. My poster is addressing the issue of antimicrobial resistance, and I'm so glad that Professor Basiro mentioned and talked about the One Health approach. AMR is a global challenge, and it's caused a lot of death. In Africa, it's estimated that about 250,000 people died in 2019 as a result of antimicrobial resistance. And to address that, a UN high-level meeting is happening in September to show you the importance that the global community attaches to antimicrobial resistance. A One Health approach is important. There have been a lot of work around human health and livestock in addressing the issue with limited attention to the environment as well as food. And food safety is quite important and could be one of the important means of transmission. We looked specifically at the fresh produce here, vegetables, especially those that are not cooked before consumption. And we saw some interesting reports in the sense that some of the pathogens, according to the World Health Organization, they released a priority list of pathogens for antimicrobial resistance this year, 2017 first and updated this year. And some of the isolates that we found were on that list. So if you want to find out more, please see me at poster 113. Thank you very much.
All right. Let's give some applause for those who did their... So once again, we're trending early. And what we did this year, in an effort to fit the schedule, is that we've combined the poster session with the coffee break. So you can get your snacks, take a look at the posters, talk to the presenters. But before we go, because we are going a little bit ahead of schedule, if you have your poster up, and we'll do this for the first 10 people. If you have your poster up, but you are not a Turbo Talk person, and you want to give us just some 2 quick things why we should come to your poster, you can come up right now.
Hello, everyone. My name is Richard Opoku, and I'm presenting on the topic Genomic Characterization of Klebsiella pneumoniae Isolates from Komfanochi Teaching Hospital. It's about isolates that have been gotten from clinical samples, and we are going to use MADI12 to confirm their identity. Thereafter, we will do sequencing. So I need you at stand 119. I hope to see you there.
For the posters. Okay, yes. To prevent the mix-up, for tomorrow we'll have another Turbo Talk session for the posters. So the Turbo Talks today are for the posters which are already up for today and tomorrow. Tomorrow we'll have another Turbo Talk session for the poster sessions Thursday and Friday. So when you are submitting your slide, make sure you mention the day that you are presenting and your poster number, which you'll get from the book of abstracts. So hopefully we'll avoid this tomorrow. So I'm going to do the wrap up for today. I think we had quite a good round of talks, more technical and some a little bit specialist, but I think we appreciate the breadth of expertise and knowledge and research that is being presented here. Tomorrow we'll kick it off with a keynote from Professor Herman Sintim, and that will be even more technical, talking about drug discovery, but that will lead into another session on immune responses and disease pathogenesis. Then we'll have in the afternoon session, we will have a change from the schedule. Our second keynote of the day will actually be Professor Nikes Ndam, because he has to catch a flight the next day, and he will switch with Dr. Janelle Binman. So I think tomorrow is going to be another very strong day of talks. In the meantime, I think we can wrap up today. Let's go have coffee break. Enjoy the posters. Please do not go home right away. As much as we wanted you to have a bit of a break, don't go home right away. Go to enjoy the poster sessions and any other housekeeping items. Andrew. Yes, and don't forget to go to the vendors. I'm sure they have some toffees or something. And then we have the WACCBIP shirts, the WACCBIP material already sewn. Okay, we're done for today.